Good morning, everybody. Respected Honorable Vice Chancellor of the University, Dr. K.N. Kattimani sir, officers of the University of Agriculture Sciences, Raichur, all the organizing coordinators, organizing co-coordinators of this 10 days webinar on role of nanotechnology in food and agriculture. We are in the last day of this 10 days program, that is 10th day. So we have only in the morning session, as we have already discussed in the yesterday. So morning session will, the first lecture will be Dr. Parsarthi is going to give a first lecture, and followed by at 11 o'clock we have a series of lectures by the thermofishers for measurement of toxicity analysis by using the high-end instruments like LCMS, GCMS, ICP-MS. And that will wind up by 12:30, and from 12:30 to 1 o'clock we have valedictory session. So 1 o'clock the whole program will be closes. Kindly be present at for 3 hours, 10 to 1 o'clock, so that we can complete all the sessions at a stretch without fail. So once again, I heartily welcome to Dr. Parsarthi for this webinar on giving the lecture on benefits of nano encapsulation. So now I would like to introduce Parsarthi for his brief biodata to our August gathering of this 10 days webinar. Dr. Parsarthi is currently doing his postdoctoral fellow at Redent Institute, Massey University, Palmerston North, New Zealand. So you know very well the most fellow scientists are none other than Dr. R. Paul Singh. So US Davis. So he is working under the R. Paul Singh as well as Arjinder Singh Redent Institute as a postdoctoral fellow, Massey University, Palmerston North, New Zealand. And earlier, Dr. Parsarthi has worked research executive at Accenture Knowledge Management. And so he has done a wonderful contribution to the masters as well as the undergraduate students for their research work and also research coordination between the students and industry collaboration, industry linkages. So he worked as a senior research fellow in the Center for Food Technological Research Institute, Mysore, India during the year 2013 to 2017. Dr. Parsarthi did his BTech Pharmaceutical Engineering and Technology, Bharatidasan University, and is completed during the year 2007. And he did his MTech in Chemical Engineering, Galgappa College of Technology, Anna University, Chennai during the year 2010. He has completed. And PhD he did CFTI campus Mysore, and he completed during the year 2017 on experimental and computational modeling approach for evaluating the absorption of nano-formulated vitamin E in a small intestine. So his major achievements are many more. So among that, he has well associated with for 20 subject experts in food technology and published a technical book in 15 months with CRC Press and generating volume of more than 230 USD and publisher to the book publisher. And he is also a recipient of SRF award for ICMR, Indian Council of Medical Research, and received 1.2 million grants. And coming to his publications side, Dr. Parsarthi has published more than 9 papers with good impact factors in international journals. As I mentioned, he has a technical book written along with Dr. Anand Ramakrishnan sir. He is a director IICPT, now IFT, Tanjavuru. So he wrote a book on food nanotechnology principles and applications, CRC Press. And almost more than 7 book chapters he has published in his credit, and all those are among 5 are in CRC publication, and 1 is TTP publication, and 1 is Willie Blackwell publications, and good contributions in terms of publications as a book chapters. Dr. Parsarthi is having main research focus on nano encapsulations, in that microfluidization, nano emulsion, solid liquid nanoparticles. And whereas in case of micro encapsulation, so spray drying, freeze drying, and spray freeze drying, he did his work in the doctoral research. And in case of analytical techniques, he used all the instrumentation, including a confocal laser scanning microscopy, SEM, TEM, high pressure liquid chromatography, particle size, DSC, and UV spectroscopy, and many more. And in terms of applications, he has developed an engineering design CAD open source 2D drawing tool and Spacecam design moderator, Autodesk Fusion 360 3D drawing tool. So, and even he knows the animal studies also, like oral intubation, retroorbital blood collection, averted gut shock, single pass intestinal perfusion. Probably he might be done during his PhD work for the toxicity analysis. And computational skills, he's having a good computational skills also, and he's expert in MATLAB also. So currently he is working or is doing his postdoctoral fellow in the University of Redent Institute under the guidance of Professor Harjinder Singh as well as the Professor Arpal Singh, UC Davis. So now with us in his busy schedule of postdoc research activities, so we have requested him and immediately has accepted for our request to giving the lecture on benefits of nano encapsulation to our participants who are there last 9 days with us, Dr. Parsarthi. Now it is over to the Dr. Parsarthi for the presentation on nano encapsulation. Dr. Parsarthi, please.
Thank you, sir. Good morning, everyone. Myself, Dr. Parthasarathi, and I like to thank Gowda sir. Thank you so much for inviting me for this webinar. So myself, I am Parthasarathi. You can hear the people call me as Partha because they can't say my name clearly. The people they shorten my name as Partha, and my in India they call me as Sarathi. Only my parents used to call me as Parthasarathi, and after that, our HOD sir called me as Parthasarathi. Thank you so much, sir. So I did my BTech and MTech from Bharadasan University and Anna University, and after that, I did my PhD from CFTRI. Then I moved to New Zealand for my postdoctoral research. Currently, I'm in New Zealand at Massey University. Recently, we have published a book on food nanotechnology principles and applications with CRC Press, Taylor and Francis Group. So in this book, this covers the entire application of nanotechnology from the processing to the final safety and efficacy studies. So this book covers whatever you need to know about the nanotechnology. So this book was edited by myself and Dr. Anandaram Krishnan sir. Currently, he's the director at IFPT Tanjavur. So this webinar is about the nano encapsulation techniques. So when able to get this opportunity, first I decided I should not come up with the different data or different graphs. I should not bore you with those data because you can get those in any of the research articles. So the one thing I kept it in my mind is you're able to get some knowledge, and you're able to, when you start your work with nano, this slide should be useful for you. So by keeping this in mind, I didn't compare any of the research article, only for few for the biology studies. So this slide, I mean, this presentation is starting from the different encapsulation techniques. So I'll talk little bit about the definition and history of nanotechnology. Then we will look into the different encapsulation techniques, how to produce those encapsulated products, like the top-down approach or bottom-up approach, and some of the liquid-based product or a solid-based encapsulated products. For example, you can produce the liquid-based product by the emulsification technique, and different solid-based product by the different drying techniques. And finally, we'll come up with the bioavailability, how you can able to calculate bioavailability with the animals, or what are the factors that affect the bioavailability of your compound. So before we start this presentation, we'll look into the slight glimpses of definitions of nanotechnology. When you ask someone asks what is the definition for nanotechnology, none can say nanotechnology is defined as, because there is no single definition or one-way definition for nanotech, because there is plenty of definitions that developed by the individual organizations that working with nanotechnology. But you can consider the particle size of ranging 1 to 100 nanometer can be considered as the nanoparticles, and the technology that's using for developing the nanoparticles as the nanotechnology. But still, when it comes to the food, even there is a debate to use till 100 nanometer to 1000 nanometer we can consider as the nanoparticle, and someone used to say until 200 nanometers you can consider as the nanoparticle. So the history of nanotechnology is started from the Nobel laureate, Professor Richard, who mentioned plenty of room at the bottom in his famous talk in Caltech. You might see many nanotechnology articles that started with the statement of plenty of room at the bottom, and even there is an unwritten rule in Nature Journal that you cannot use that statement when you start your article, and even you cannot find such in many of the Nature published articles. But the word nanotech was coined by the Japanese professor in 1970s for his work on the basic concept of nanotechnology. But until we reach the 1980s, there was not much contribution in nanotechnology until the people find the scanning tunneling microscope in 1980s. After that, the role of nanotechnology or the contribution of nanotechnology become enormous. Now we are exploring the possibility of bringing nanoscale machines or nanocards. Um, the nanotech, we used to think it started from in the 1980s, but when we look into the history, the Ajanta Ellora caves, the paintings are still maintaining its color. The researchers find the presence of nanoparticles, carbon nanoparticles, in those paintings are maintaining its color because in those periods there is no such synthetic chemicals or any other paint materials for those paintings. And still we didn't know how those nanoparticles are produced by our people, and what are the techniques they have used. The other interesting one was developed in the Roman period in the 4th century, that is the Lycurgus cup. It's still preserved in the British Museum. The beauty of the glass, it normally looks green, but when you turn the light on it, it turns to the red color. It's really surprising how a glass can be dichromatic green. And the later researchers found it's the presence of the gold nanoparticles in those cups that reflects the color into bright red when you pass the light. So when I was working for the food nanotechnology book, I came across the articles on Damascus sword. This is the dated back. We have used, our soldiers have used until the 18th century. Well, these swords has a very sharp edge, and even it still remain its same sharpness. Even you can find in the many museum, and even I have read the article about the Tipu Sultan war, who have used these such Damascus blade. So these blades can able to cut even a silk kerchief in the air into the fine pieces, which is certainly not possible with any of the any of the swords. So the researchers found these swords contain the carbon nanotubes and carbon nanowires. These swords are developed by our Indian bladesmith, but they can't able to. I mean, the European bladesmith, they can't able to replicate the same sword in any other countries. So the whole world is crazy of nanotechnology. Even in the research, around 60% of American population have heard or known about the nanoparticles or nanotechnology. Even there was a period, the article can get easily published when you have worked with nanotechnology. Even I mean, I'm saying the period of from 2000 to 2010. If you have any article with this with the nano work, or you have a small part of work with nano, it can get easily published. So what makes the thing crazy for nanotechnology? The only thing is the one of the thing is the surface area that you're going to receive after particle reduction is enormous. You can imagine a spoon of nanoparticle can occupy a whole football ground, such a huge surface area it can able to provide. So the way you can think the application of nanotechnology in painting chemical reaction with respect to the surface area, if you're having bulk particle for a chemical reaction, the same bulk particle in the breakdown into nanoparticle for surface reaction, definitely you will get a huge change with respect to the reaction rate. So coming to the our topic of interest, nano encapsulation. The word encapsulation means literally you are protecting your compound of interest with some wall material. For example, you have the very heat sensitive wall material like fish oil. So you can't use the fish oil, and even the fish oil is highly order very the bad order that make unpleasant to the other person who is sitting in the room. So when you are encapsulating the fish oil, you are able to mask the taste and you are able to increase the increasing the stability of the material. So the need for the encapsulation comes when the product is easily denatured by the temperature processing conditions, or even during storage, or when you are keeping in the product in the cells for long period, or when you consume in the gastrointestinal tract, it may get denatured. So the nano encapsulation, you are protecting the bioactive compound with materials on two conditions. Either the bioactive compound should remain in the nanoscale, or the entire system should be in the nanoscale. We will see next how to fabricate the such nano encapsulated products. As you all know, there are two approaches for making a nano encapsulated product. One is the top-down approach, and in the bottom-up approach. In the bottom-up approach, the materials arrange themselves into nanoscale by self-assembly and self-organization of molecules. This bottom-up approach is influenced by many factors, including the pH, temperature, concentration, and ionic strength. In the top down approach, the excess amount of energy is given to the bulk materials and break into nanoscale. In a layman term, I can say if you glue the atoms by physical and chemical forces and make a nanoparticle, it is called the bottom up approach. On the same way, if uh, you take a hammer and break the bulk material and bring the nanoparticle, it is the top down approach. So in nano encapsulation, you can use for top-down approach like the emulsification technique, or emulsification solvent evaporation, or ultrasonication technique. Each technique in the top-down approach that has the different principles. For example, in ultrasonication, which works based on the cavitation force. In the emulsification by high pressure homogenizer or microfluidizer, you're going to use the enormous energy on your system. In the bottom-up approach, as I mentioned, it works based on the self-assembly of atoms or the clusters. So you need not give any such external energy, but it's too hard to use for the industrial scale because of the yield that you're going to receive. The bottom-up approach includes the nano precipitation, co-observation, and inclusion complexation techniques. We're going to see only the emulsification and nano precipitation. In the emulsification technique, we're going to add two immiscible phases and bring into the one phase, because most products, the food products that we are taking in daily life is a product of emulsion. So even my first research, I started developing the vitamin E nano emulsion. Emulsion means it has oil phase, water phase, and a surfactant. In oil and water nano emulsion, the oil droplets are dispersed in the water phase, and the oil droplets should be in nano size. Emulsification is a process by which you develop a system of two immiscible liquids, usually oil and water, one of which is dispersed as a small droplet in the other, like oil dispersed in water or water dispersed in oil. The typical size range of nano emulsion is 500 to 200 nanometers. For instance, the oil in water nano emulsion, the oil droplets have the particle size of 100 to 200 nanometer, and the surfactant, you can have a chance to use either the a synthetic surfactant or a natural surfactant. And I would say in recent days, there is an interest on natural surfactant for using your emulsions. So through different emulsification technique, it is possible to load or encapsulate a high concentration of nutraceutical or bioactive compounds. Also, the nano emulsion can be used in food through different fortifications. You can encapsulate the lipophilic bioactive compounds like the beta carotene, carotenoids, dietary fats by oil in water emulsions. For water soluble bioactive compounds like polyphenols, you can encapsulate by water in oil emulsions. So, when you look into the emulsification technique, we look into the some of the high energy emulsification techniques. So, who work with the nano emulsion, they definitely come across these two emulsification. I mean, the high energy process. One is the high speed homogenizer, and other is the microfluidizer. You can develop the emulsion by high energy or low energy. So in high energy, you're going to give a huge energy that you can compare with the elephants. I'll come with the calculation at the end of this slide. So the high speed homogenizer is the most convenient one when you prepare for a emulsion, but the stability of the emulsion through the high speed homogenizer is always questionable. So most researchers, they start their pre-emulsion with the high-speed homogenizer, and then they will move to the microfluidizer or high-pressure homogenizer. In the microfluidizer, they use high energy. The intensity of the pressure that act on your emulsion will break the oil droplets into the fine nano droplets and maintain its stability in the nano emulsion. So you can able to provide even 20,000 psi pressure, but usually the people, we don't go beyond 10,000 or 12,000 psi because the heat that generate in the emulsion is it's too hard to reduce that, and we may not have that facility to reduce that heat generated. So the rule of thumb is, if you work between the 6,000 to 9,000 psi, that will be quite enough to make an nano emulsion. So how do you relate the PSA? What does the PSA means? How can you imagine 1 PSA, 2 PSA? So for example, yeah, elephant of 2,000 kilos that's standing on a 1 meter by 1 meter slab. So the pressure that exerting on the foot is 116 PSI. So here the 6,000 PSI means nearly 50 elephants that's standing one above the above. The final pressure that you're going to receive will be equivalent to 6,000 psi. So once you develop the nano emulsion, do you think the emulsion will be stable for a long period? So what are the ways it can able to destabilize? So the emulsion you prepared, you can't imagine that it will be stable for a long period. It depends on the type of emulsifier you have used, the concentration of emulsifier you have used, or the energy you have used for developing the emulsion. Usually the emulsion can be destabilized by 5 different mechanisms like flocculation, coalescence, oswald ripening, creaming. So in flocculation, the droplets, the fine droplets that stick together and remain as a collection of smaller droplets. So when you look into the microscope, you can see the clusters of small droplets in the in your slide. In coalescence, these small droplets are merged and it form a larger droplets. So when you prepare an emulsion before going for a product, the people, I mean the industrialist, will look into the stability mechanism on how it able to destabilize for the different storage conditions. So the next one is the bottom-up approach, is the nano precipitation technique. Nano precipitation is one of the economical method that literally you need not need any such huge sophisticated equipment, because in the previous slide we have come across the microfluidizer. I think the price of microfluidizer in India is around 40 to 50 lakh, probably in 2000 14 or 15 when I have when in my lab we have purchased. So in nano precipitation, you don't need such a huge sophisticated equipment. This technique is mostly preferable for a lipophilic compound. In nano precipitation, you are adding an organic phase into the aqueous phase. Basically, the organic phase contains a film forming material, the lipophilic bioactive compound like curcumin or surfactant and the organic solvents. So you are adding the organic phase drop by drop to the aqueous phase, and you remove the aqueous phase by the solvent evaporation, and finally you will end up with nanoparticles. Even I come across a research article that they are able to achieve 99% of encapsulation efficiency, which is too tough even when it comes to the nano encapsulation. So even you can able to combine the freeze drying technique in this nano precipitation, and be able to achieve the fine porous particles. So until now we have seen the different emulsification techniques. The products of the emulsification would be the solids. I mean the liquid products, but the liquid products has the problem in during the storage and during processing. So once you convert those liquid into the dried solid products, it will be easy for you to take it to the consumer hands. Literally, we're going to see around 4 drying techniques, how we able to achieve the nano encapsulated products through those drying technique. So the first one is the spray drying, which we are using more than 100 years in India. The frequently used consumer products like the milk powder, instant coffee, even our washing powder or the products of the spray drying. In spray drying, you are feeding the liquid and atomize in the hot environment and evaporating the liquid phase and ended up with the solid particle. How do you say the product that you are achieving in the spray drying as a nano encapsulated product? Because definitely the end product you can't find as any nanoparticles in that, because the after atomization and the final end product will be around 200 to 300 micrometers. Technically, the liquid phase contain the nano emulsions, and in the product, the nano droplets remain in size even after drying. But the problem is you cannot go for spray drying if your product is easily oxidized or thermal sensitive. For example, if I have a fish oil which will oxidize very easily, I definitely cannot go for the spray drying technique. So when you look the spray dried products in the scanning electron microscope, you can find so
Hello, Partha.
sir, 1 minute, I'm coming back.
Okay, no problem. No problem. You can have water, Partha. No problem.
So in spray drying, the product you're going to get, if you look into the scanning electron microscope, you can find the spherical particles with so many dents and surface irregularities. So that such dents will definitely affect your dissolution properties. In freeze drying, that will be an effective alternative for the spray drying, because in In freeze drying, literally, you don't need such a huge temperature. But the problem with the freeze drying is it will take a long time, definitely a long time to get the product. It will take even 24 hours. Even the freeze dryer at Massey University will take nearly 2 days to get the product. So you need to run the compressor and the refrigeration unit for 24 hours to 36 hours. And when you look into the product, you can find the flakes and the porous structures on your product. So these porous structures will help you in the dissolution. So what do you think when you're able to merge the advantages of these two drying techniques, like the spray drying and the freeze drying? The spray drying, you're able to get the spherical particles. In freeze drying, you're able to get the porous particles. So you will ended up with the spray freeze drying technique. In this technique, you are literally spraying, atomizing your liquid droplets into the liquid nitrogen. So which once you atomize the liquid nitrogen, those droplets will turn to the will maintain its shape, spherical shape, and it will frozen in the liquid nitrogen. Then you can take the particles to the freeze dryer, and you can remove those frozen ice crystals, and finally you can get the spherical particles. So when you have the when you take the freeze dried products to scanning electron microscope, you can find the particles will be in spherical shape. And you can find the numerous porous structures in those particles, that is due to the sublimation of ice that happen in the freeze dryer. So the main benefit of the spray freeze drying is you're able to get the spherical particles with numerous pores that will help you in the dissolution. The other one is the electrospraying technique, which is also called the electrodynamic spraying. It is capable of producing the nanodroplets by means of electric field. In this process, the high electric field is applied to the needle, and then the charged liquid break into the nano-sized droplets and settle at the ground collector. Unlike the conventional process in electrospraying, you can perform at ambient conditions. I mean, you don't need any separate condition like in spray dryer. The entire temperature will be around 100 plus degrees Celsius inside the spraying chamber. But electrospraying, you can maintain the same temperature, and you need not change any pressure. So it will be helpful for you when you are using a very heat sensitive biomolecules or even the live cells. And the other biggest advantage with the electrospraying is the dissolution material you are using is just water. You don't need any other solvent to get the nano encapsulated product. But the main disadvantage with the electrospraying is the yield that you're going to achieve. It will take nearly only a few milligrams or few grams of sample when you run the machine for 1 or 2 hours. So it is hard to think to get the massive amount of products through the electrospraying technique. So why do we nano encapsulate a powder? What you're going to get what are the key benefits that we're going to get with the nano encapsulation? Yeah, so mainly the nano encapsulation able to protect your material in the gastrointestinal tract, and it helps you in increasing the shelf life of your material. That means you can store your product for longer time in the supermarket, and it helps to mask the odor or taste of the bioactive compound, like especially for the fish oils or omega-3 fatty acid. Even I do remember when I worked in CFTRA, one of my colleagues used to work with omega-3 fatty acid. Once you open the bottle, no one can stay in the lab for a week. The smell of omega-3 fatty acid will remain in the lab. But when he bring it to the nano emulsions, the entire smell will be the odor is masked, and he achieved nearly 100 days stability, and the paper is published in the Food Chemistry. You can find it there. So when you are nano encapsulating, you are literally increasing the surface area. That means you are improving the overall bioavailability of compound. And finally, you can able to trigger the release of your biotic compound when you are consuming it. You're able to provide the targeted drug delivery like that. You can able to work with that. So the whole product, I mean, the whole nano encapsulation work of finally working towards the bioavailability. The term bioavailability, the whole in the food industry are crazy about the bioavailability, how we can increase the bioavailability, what are the ways we can able to increase that. Even you can see the some of the health drink products are specially targeted towards the bioavailability. Bioavailability is defined as the portion of nutrients that's getting absorbed in the blood. For instance, you are taking a 10 milligram of nutrient and 1 milligram of nutrient is finally absorbed in blood, then the approximate ratio would be the bioavailability. But when you take a nutrient, it has to come across a series of barriers, starting from the intestine. Then it has to cross the gut wall metabolism. Then it has moved for the to the liver. The finally it metabolizes in the liver. The final amount only will come into the blood circulation. The food industries are working on the ways to improve the bioavailability. One way is the nano encapsulation that we have seen. Even I have worked with the nano emulsions, and I vary the size of the nanoparticles from the 100 micrometers to the 200 nanometers to the 1000 micrometers like that. And I have checked how the bioavailability will differ for each nanoemulsion for the same content of vitamin E. So in the bioavailability study, you can work with three different techniques. One is the in vivo technique, in vitro, and in silico. In the in vivo, you are working with the animals. In vitro, you are working with the instruments, and in silico, you are working with the computer simulation. At New Zealand, I'm working with the in vitro system. That means I'm developing an artificial simulator for the digestion work. You may come across the articles like human gastric simulator. So that will mimic the entire gastric condition in artificial system. You need not go for the animals, and you can work completely with the in vitro system, and that result is acceptable according to the FDA. So we'll see only the in vivo study, how we can able to calculate the bioavailability through animals. So in this study, the people use the small animals like rats, mice, rabbits, and pigs for the research. And they wait for a few hours, and then they collect the sample, and they analyze the content, how much the sample, the nutrient, the present in the blood. Usually this study, the bioavailability study, will be, you can say, as the pharmacokinetic study. So that means what the drug that does to your body. In food, we consider only the three parameters that considered to be very important for the bioavailability. One is the Cmax, that is the maximum concentration. Tmax, the time at which the maximum concentration occurs, and AUC is the area under the curve. So in this curve, you can see the I given three different emulsions. One is the nano emulsion of size 250 nanometer, and conventional emulsion of 600 nanometer and 1200 nanometers. All has the same quantity of vitamin E, and we have calculated how much vitamin E that present in the blood after 6-hour period. So you can see the Cmax for the nanoparticles is 11.25 milligram. That means that is nearly threefold increase in bioavailability when you have given a nano emulsion than the conventional emulsion. though there are so much of benefits for the nano based products, but still it is necessary to see the how the nanoparticles that interact with our human biological system, because the nanoparticles has a high surface area in body. That means it has the surface energy is also increasing. So when it enters the body, it able to attract the protein, and it forms a complex structure called protein corona. In research, nearly 350 proteins can be interacted with the nanoparticle in the blood plasma. So when you take a nanoparticle, there is a chance for the nanoparticle to cross your biological barriers like the blood-brain barrier, and it able to interact with it. So before the nanoparticle comes into the product, into the consumer hand, it is necessary to have a deep understanding of nanoparticles, so that we can able to get the safety, maintain its safety. So the nanotechnology has both merits and demerits. So we need to go for analyze the complete analysis of the uh, product, and it has to be reached the consumer hand. So if you ask, are we still using the nanoparticle, does the nanoparticle based products has reached the consumers in India? If you ask that, yes, I would say yes, because these are some of the products that has reached in India. Uh, uh, Even I would say these products are boldly they mention the word nano in their labels. Even in a recent regulation studies, if you use the nanoparticle, you have to mention nano in your label itself. The IFCO is it's a government-based fertilizer company that's working with enormous amount of work on the nano-based fertilizers. So soon we can look some more products on the nano encapsulated, especially in the healthcare and in the nutraceutical products. Thank you.
Well, Dr. Parsarthi, sorry for some problems during the talk. I think your throat is not. Yeah. Can you stop sharing, Partha?
Yes.
Yeah. Thank you very much, Dr. Parsarthi, for your very nice systematic presentation in the field of encapsulation and how we are going to use it in the field of food and agriculture and in general too also. So we can have some question and answer if you have a time, so that we can maybe another 10 minutes or 15 minutes. Is it okay, Dr. Parsarthi, so that we can have maybe some questions? Is it okay? Dr. Parsarthi, can you hear me? So can we start for the question and answer session for 5 minutes? Is it okay? You are unmuted, Parsarthi, sir. Sir, can you unmute yourself?
Yes, sir.
Yes, sir. Sir, is it okay for 5 minutes question and answer session?
Definitely, sir. Definitely. I'm happy to help. I'm happy to answer, but I'll be more comfortable if I get the questions in the written format. I can able to answer them.
Sure, sir. Sure, sir. Maybe one or two questions. We'll take
Yes,
it,
sir.
and then, yeah. So chat box is open for audience for 10 minutes, because actually Parsarthi is not feeling well, I believe, but even we are troubling you, sir. Sorry for the inconvenience caused due to.
So certainly not, sir. I'm happy to.
Okay, fine, sir, fine, sir. Sir, for encapsulated material, I'll repeat, for encapsulated material, so how we can go for the analytical part, that is scanning electron microscope?
It's a scanning electron microscope. You're able to see only the how the surface will looks like.
Yeah. So
so you can't quantify anything with the scanning electron microscopy. Only thing when you are going for a dissolution characteristics, or how it looks, you can see in the scanning electron microscopy.
Okay. There is another question. Okay. I'm not getting any questions from the chat box, but both YouTube as well as chat box is open. Okay. Let me see in my personal chat. Sir, is the nano encapsulated food materials available in India and other parts of the world?
Yes, sir. It looks like that because when I looked in the nano in the website, I had seen some nano encapsulated curcumins are available.
Okay, nano encapsulated. Okay. But in India, sir, or in?
No, it's in India. In India.
Okay, sir. Because why the participants are under this kind of questions? We heard that during the couple of presentations, still there is a pending with the regulations
Yes.
in the Indian government. But the people are claiming because this is encapsulated with this, but we don't know what is the toxicity about all those things.
True, sir. True. I agree with you, sir.
Yes, sir. Sir, plant essential oils are best products as medicine and agricultural purposes. Can we nano encapsulate the essential oils, sir?
Sir, it has been already encapsulated. It is under the research work.
Yes, sir. Even like our omega-3,
Yeah.
yes, sir, it's already encapsulated and they're doing. Even fish oil also they are encapsulated.
Yes, sir. Yes, sir.
Sir, how bioavailability of nutrients varies from direct consumption and nano encapsulation?
Yes, sir. I'll give up with the example. For example, we take beta carotene that's present in the carrot. So when you take a beta carrot, consider it will get absorbed a very little amount. So if you have a recommended daily intake of 30 micrograms, so like you need to take nearly 1 kilogram of carrot. That's certainly not possible for anyone to take. So if you take a nano encapsulated beta carotene, you can take the same 50 microgram, and the entire 50 microgram can able to achieve in the systemic circulation, sir.
Okay. Sir, can we make nano uniform individual structure materials from organics for food application?
Sir, certainly I don't know. That's beyond my scope.
Okay. So it is possible, sir, but only thing is the conversion formulation may be different. Sir, how nano encapsulated seeds will give protection against diseases? How nano encapsulated seeds will give protection against diseases?
Sir, no, sir. I haven't worked in this. I can't able to give any comment on this, sir.
Correct, sir. Correct. But generally, we do the encapsulation with different materials for the seeds. So that will protect as an antimicrobial or antifungal, like that they are using for the seed treatment, nanomaterials.
Okay, but the seed is already in a micrometer range or millimeter range. How do we claim that as a nano encapsulated? Probably the encapsulated layer may be in a nanoscale.
Yes, sir. Probably the encapsulated layer may be nanoscale, sir. So they will go for like electrostatic spinning or something like that, sir. Sir, nano encapsulation is possible with a single cell protein, that is spirulina, since it is having a fishy smell. Nano encapsulation is possible with a single cell protein for the spirulina, since it is having a fishy smell. It's bad smell. Or,
okay.
Can it be possible?
No, sir. I have to look into the literature.
Okay. Because generally the encapsulation to avoid those kind of aroma and flavor.
Yeah, that it will happen, but I'm not sure with the single cell protein. Because I have worked, I mean, my colleague have worked with the fish oils, and in our group we have worked with the vanillins. So it has produced the good masking the aromas over there.
Okay, sir. Some of the people predict that in future we will have a tablets of nutrients, wherein there will be no need of bulky plate foods. Is it be possible?
Uh, at the moment, we do have tablets of nutrients. One of my colleague is having the problem with the intestinal villi. She doesn't have villi, so she is spending her days only with the tablets of nutrients. I mean the micronutrients, whatever she's having, that she's taking the.
Okay. So there may be possible, but it is not exactly substitute of what the bulky food. Am I right, sir?
Yeah, but if you're asking me personally, I would say what is the need for the tablets?
Correct. Correct. Sir, since you are working on animal model, what is your opinion on safety aspect, especially on genome toxicity?
We work with animal model mainly on the food digestion. We haven't come across any such genetic levels. So I'm not the right person to answer on
Okay.
this question.
Okay, sir. Sir, what are the nano food or vitamin capsules available in market? So already there are a couple of things are available. But
Yeah. I recommend there is a website which collect all the data of nano based products. So I recommend Chandra's sir to look into that website. He can able to get the entire information.
okay, sir. What is that website, sir? Can you tell?
Sir, once this complete, I can send you the mail, sir.
Sure, sure.
It's like nano.org related to that.
Is it nanorex, sir?
Sir, I have to cross check with my system and I'll come
Sure,
back.
sure, sir. Sir, how the nutrients of encapsulated products in dairy foods are short shelf life products where cultures get multiplied?
So the dairy foods like the probiotics.
Yeah, yes, sir.
So I didn't understand this question.
So how the nutrients of encapsulated products in dairy foods are having a less shelf life products where cultures get multiplied.
Yeah, I can say about probiotics because we have worked with the lactobacillus strains. So the concept is you are giving the symbiotic material that able to maintain its growth inside the encapsulated product.
Okay. Sir, how single fold is better than three-fold nano emulsion? Single fold is
Okay,
better.
okay. Actually, the single fold, the fold is to compare your final result. So if you're taking a conventional emulsion, you are getting a 10 microgram in your blood. If you're taking a nano emulsion, you will get 30 microgram in your blood. So you can say the 30 microgram will be better for your. So he's asking about the. So you are taking 30 micrograms that will come into your blood. That means it has a better future than the conventional one.
Okay, sir. Sir, what is the size range of encapsulated materials that one can call them as a nano?
Yes, sir. When it comes to the encapsulated product, your bioactive compound that present inside the material is maintain its nano size. So, for example, 200 micrometer, 200 nanometer will be sufficient.
Okay.
Because certainly we can't recommend the people to go with less than 50 nanometer because there is no evidence regulations based on this.
Okay. Sir, almost we have done the question and answers in the chat box of my personal chat box. Thank you, Dr. Parthasarthi, for your nice presentation, and that too. There are a couple of questions also is joining with us. Can we nano encapsulate microorganisms?
Here we need to consider the microorganism. It's certainly not in nanoscale, but you can make a layer above the microorganism in nanoscale range.
Okay, okay, sir. Uh, Thank you very much, sir. If any questions and answers is there, definitely I will just pass on to you for the further clarifications to our fellow participants. Thank you very much, sir. On behalf of the organizers, on behalf of the university, and on behalf of all my participants, I sincerely extend a thank you for your kind presence. Probably it is late evening for you, I believe so. So thank you very much for the morning session. Thank you, Dr. Partha.
Thank you so much, sir.
Yes, sir. Yes, sir. So we'll keep in touch with you, Partha, for any clarifications in due course of time, for any clarification from our speakers or any clarification from our fellow participants. Thank you. So we
Sure.
have in the second session, as I discussed in the yesterday also, and even in the morning also, we have a bunch of speakers from Thermo Fisher for toxicity evaluation by using the high-end equipment like LCMS, GCMS, ICP-MS. Dr. Ramesh, can you hear me?
Yes, sir, I can hear you.
Yeah, fine, fantastic. I think Sarvendra is also joined. Dr. Sarvendra, can you hear me? Sarvendra Pratap Singh? Okay.
Yes, sir, he's along with me, so he can hear you well.
Okay, fine, fantastic. So shall we start, Ramesh, if you don't mind?
Yeah, sure, sir.
Because we are running out of time in the morning itself. Because there is another program is hanged with our Director of Education as well as the Director of Extension, that is the PMO inauguration of Central University. That is going to be taken by 12:30. So we just pre-funded our valedictory program at 12 o'clock instead of 12:30. Once the valedictory program completes, then we can continue our session for the further speakers. We'll see how best can be, things has to be done. Is it okay, Ramesh?
Yeah, perfectly fine, sir. No problem.
Yeah, thank you. So I would like to first introduce Ramesh Azad. He is basically an application development scientist, food and beverages. He has completed his Masters of Science in Biotechnology. He is having more than 11 years of experience in the field of LCMS, GCMS, and various development through different segments like food, beverages, pharma, and environment. He has extensively working with the different pesticides applications, veterinary drugs in various food matrices, and its validations. So we have with Ramesh on the topic on LCMS or MS, a tool for trace level quantification of contaminants in food matrices. Now I request to Ramesh, please. Yeah, Ramesh, please.
Yeah. So if you can confirm, sir, my slide is visible?
Yes, sir, it's perfect.
Yeah. So thanks for the nice introduction, sir. And today I'm going to talk about the basics of liquid chromatography and tandem mass spectrometry. And we are going to touch upon some of the applications of LCMS and mostly on pesticides and antibiotics. So we have a team of Sarvendra and Suresh, along with Dr. Dashrath, who is heading us. And they'll be talking through about GCMS and ICP-MS. So my topic would be on basics of LCMS. So the agenda is like, we are going to talk about the basics of liquid chromatography. I know the time is very short, so I'll be doing it a little fast because the LCMS technique, it is not very straightforward to understand in one go. So, but still I will try to make it simplify for you people. So we'll go for the basics of liquid chromatography. I'll just put my laser pointer here. So basics of liquid chromatography, then what are the components of HPLC and UHPLC, basics of mass spectrometry, working principle of LCMS-MS, then different ionization technique which we use in case of LCMS-MS, then the mass analyzer, and then maintenance and how to improve your method performance, especially for LCMS-MS and different food safety applications. So we are going to see pesticides, antibiotics, and mycotoxins. So directly we'll go to the history of liquid chromatography. So there was a Russian scientist called Michael Sweat, and he has done the experiment on the column in which there was a calcium carbonate filled with alumina, and he poured the green extract of plant, and later on he saw that different bands are there. So when he poured the green extract of a plant and through the column of calcium carbonate and alumina, and after some time he could see some different bands, and later on it was concluded as a chlorophyll pigments. So this was the starting of chromatography. So it was like we are able to separate different components, different organic components using a stationary phase made up of calcium carbonate and alumina. So later on, it was like developed like different stationary phases are available, and nowadays we use silica as a base material for LC stationary phase. So if you see the HPLC, so HPLC long form is high performance liquid chromatography. So it is an analytical technique which is used to separate, identify, and quantify each component in a mixture. So each component it's an organic material. So the mixture is separated using a basic principle of column chromatography and then identified and quantified by the spectroscopy. And there is a computer which is attached to the machine which will help you to quantify and review the data. So if you see here, it is like I have a different components. I have a first mixture of different organic compounds. And then I have a column here, which is made up of silica or alumina. And when I pour the sample here, so according to the interaction of different organic compounds with the stationary phase, different compounds will elute. So for example, if my stationary phase is like non-polar and my mobile phase is polar, and if my compound is polar, so obviously it will not retain more because my mobile phase is polar and my stationary phase is non-polar. So the compound which is polar in nature, it will not retain much and it will elute fast with the polar mobile phase. Whereas the weak or mid-polar compound will retain more as compared to yellow compound, and it will elute later. And the last one is the blue compound, which we can see it's a non-polar compound. So the thing is like when you have a non-polar stationary phase, the polar compound will elute first, then your mid-polar compound, and at the last, the non-polar compound. So overall, if you see the chromatographic process, so it is like we have a column here, which is made up of silica, and we call it as a stationary phase. Then there is a continuous stream of mobile phase. So mobile phase, since this is a liquid chromatography, so it's a liquid, whereas in case of GC, it will be a gas. And after this, when we inject the sample, so whether it is a manual injection or automatic through auto sampler injection, so if these are the three compounds, that is yellow, red, and blue, so it will be separated like this. So on the basis of their interaction with the stationary phase and the mobile phase, the compounds will elute and ultimately get detected by a detector. So I'll not touch upon this slide much. These are the different columns. So generally we have a PrEP HPLC, which has a little bit higher particle size than a conventional HPLC, and then UHPLC. So UHPLC is like ultra high performance liquid chromatography. So the PrEP HPLC, if you see like any manufacturing industries, like if you happen to see a Jhandu factory, so wherein they separate the different components, different active ingredients like menthol. So they actually separate it through a bulk column. So these are the big columns. So they separate it and then they blend it together to form a particular product. So the aim of this PrEP HPLC is to separate the different compounds. Whereas in case of HPLC and UHPLC, it is like to separate and identify and detect those compounds at a trace level. So maybe it is at percentage level, ppm level, or sometimes ppb level as well. So as compared to HPLC, UHPLC gives a more sharpness, more accuracy, more resolution as compared to HPLC. So nowadays people are using a UHPLC for the faster separation and the better resolution in chromatographic process. So the components of HPLC. So as I mentioned, it's a liquid chromatography. So we require a mobile phase in the form of liquid. So it can be acetonitrile, water, and methanol. Then we require a pump. So the function of pump is to pump the mobile phase. Then there is an injector. So injector, it could be an auto sampler or manual. So it will automatically take the sample, or if the manual injector is there, we need to inject the sample manually. Then this is a heart of your HPLC. So this is a column in which the separation will happen. So all the compounds will separate inside the column. So once we have a sample injected through the injector onto the column, there will be separation of different compounds onto basis of their interaction with the mobile phase and the stationary phase and the detector. So detector are different kinds. So conventional detectors are like UV, PDA, RI, and the most advanced and accurate detector is a mass spec. So that we are going to see in my later slides. And then there is a computer which is connected to the system, in which we are going to run our samples, see our samples, what are the different compounds are present, then we can quantify the samples, and finally we can report out. So this is a basic of diagram of HPLC. So if you see a product, like that was previous slide was a conventional diagram of HPLC, but if you want to see in the form of products, it look like this. So if you see, this is a column compartment. This is the mobile reservoir where we keep the mobile phase, that is any liquid, methanol, water, and acetonitrile. Then there is a detector. This is the auto sampler and the pump. So if you open the machine, so if you see here, this is a mobile phase, this is a detector, this is auto sampler. So small vials, if you can see here, so we put the sample into the small vials, and this is the injector port, which will automatically take the sample. And this is the pump. So the function of pump is to pump the mobile phase from here. So it will pump the mobile phase at a particular pressure, and this is a column. So the flow is like your mobile phase is here, your sample is here, your pump is here, your column is here. So pump will start pumping the mobile phase. Auto sampler will automatically take the sample, inject onto the column, and the separation will happen, and it is connected to the detector. And after that, you get the signal in the form of chromatogram in your PC. So this is the column, and if you want to have a more than one column at a time, so you can have a different column space wherein you can put more than one column. So if you see the column chemistry, so there are different HPLC modes. So normal phase chromatography and the reverse phase chromatography. So in case of reverse normal phase chromatography, the stationary phase is polar and the mobile phase is non-polar. So in case of normal phase, you will use non-polar mobile phase like hexane or heptane or ethyl acetate, since your mobile phase is, since your stationary phase is polar. Whereas reverse phase chromatography, so 90% of applications in case of HPLC is using a reverse phase chromatography only. Very few applications are there which we use a normal phase chromatography. So in case of reverse phase chromatography, the stationary phase is non-polar and the mobile phase is polar. And if you see here, so this will clarify more. So it is like if the stationary phase is polar. So if my stationary phase is polar, and if I introducing the sample, so for example, LO is a polar compound, so it will retain more. So like attracts like. So here it is like polar stationary phase, polar compound, it will retain more onto the column, and it will elute later. Whereas a non-polar compound, which is blue in color, which will not retain more, and it will elute faster than the other compounds. And similarly, the opposite way in case of reverse phase chromatography, wherein your non-polar compound will retain more, whereas polar compounds will elute fast along with your mobile phase. So there are different versions of HPLC, isocratic HPLC and the gradient HPLC. So if you have a mixture of one or two compounds, then in that case, isocratic HPLC will work. So what is meant by isocratic HPLC is like when you have a mobile phase, like for example, if I have A and B as a mobile phase, which is having a methanol and water, and suppose I have a runtime of 10 minutes, so I'll keep the mobile phase composition same throughout the run. So 50-50 water methanol, it will be there for 10 minutes. Whereas in case of gradient HPLC, we change the mobile phase composition throughout the run. So like from 0 to 1 minute, we are keeping 99% of aqueous. Then we are at 1 minute, we are keeping 75% of your aqueous. Then we are gradually goes on increasing the organic content, that is methanol. So what is the advantage? So if you have a compounds, like what we do in our lab is like we do close to 300 pesticides in 15 minutes. So isocratic HPLC, if I had to say, separate all those compounds through isocratic method, so it will take a long time. It may take 100 minutes or 50 minutes, and also the resolution will not be that much good. But in case of gradient HPLC, I'm able to separate 300 pesticides using a gradient HPLC in 15 minutes. So that's the advantage we get over isocratic HPLC using gradient HPLC. Then there are different pumps. So there is a quaternary pump and the binary pump. So in case of quaternary pump, there is a single pump which will pump 4 mobile phase at a time. Whereas binary pump, there will be a separate pump to pump the mobile phase. So it's a high pressure mixing. As such, in case of contaminant analysis or toxicological analysis, we don't find any difference in analysis through the binary pump or quaternary pumps. It is almost similar. Only some pharmaceutical critical applications, they use sometimes quaternary pump or a binary pump. So they have their own validated method. So if they don't want to shift onto other methods, so they keep it like a quaternary pump or a binary pump. So talking about detectors, so these are the conventional detectors which has a capability to identify and quantify compounds at percentage and ppm level. So this is like UV detector. So, and then the diode detector. So UV will be have a UV range only, whereas diode detector will have a UV visible both the range. Then the refractive index detector. So wherein like sugars, sucrose, glucose, like carbohydrate analysis, we use a RI detector. It is a universal detector. We call it as a, and then the fluorescence detector. So which is the most like, if you compare with these three conventional detectors, the fluorescence detector is the most sensitive detectors. That too, especially for aflatoxin analysis and polyaromatic hydrocarbons analysis. So this sensitivity of fluorescence detector can be compared with the mass spec analysis, but this fluorescence detector giving a sensitivity is limited to particular molecules like pH and aflatoxins. And the most advanced and the accurate detector till now in the industry is the mass spec, which we are going to study in my later slides. So if you look at the applications of HPLC, so there are various applications of HPLC, that is in pharmaceutical. As we know, they test the API, then genotoxic impurities and everything in pharmaceutical, then forensic, clinical, environmental, food, consumer products, and etc. So we'll go through the MS now. So MS is like, as I mentioned, it's a very accurate detector till now in the industry. So what it does, it identifies the compound onto basis of mass to charge ratio. So like every molecule, every organic molecule in the universe will have its own mass. And that is a correct way or accurate way to detect a particular organic molecule in your sample. So what it does is like every compound will have its own mass, but to be detected by a detector like MS, mass spectroscopy, so it need to be a charge. So that is the reason we use a different ionization technique. So it cannot be detected at its neutral state. We need to make it charge that particular molecule, and then we can detect it by mass spec. So that is the reason I said like we detect the compound on the basis of their mass to charge ratio. So LCMS, if you see here, so it's a hyphenation technique. So when we combine the LC with MS, it becomes a hyphenation. So what we are doing, we are combining the separation power of HPLC with the detection power of mass spec. So it's a hyphenation technique. So when it is connected to a GC, it called as a GCMS. When it is connected to ICP, it is called as a ICPMS. All are hyphenation technique. So here it is a HPLC, UHPLC, and this is a mass spec. So how the mass spec works is like, as I mentioned, it detects a compound on the basis of their mass to charge ratio. So first of all, we need to produce the ions. We need to make them charge these molecules. So for that, we have a ion production unit. So we call it as an ionization chamber. Then ion optics. So once the ions are formed, they need to be moved towards the next part. So that is a quadrupole. So quadrupole is a heart of your mass spec. So the one by one, I'll tell you the function. So ion production, there will be a production of ions. Then ion optics, which will move the ions towards the quadrupole. Quadrupole, it will select a particular mass. So what it will do, for example, I want to identify out of 100 compounds, I want to identify 10 compounds of a respective masses like 100, 200, 300, 400. So I'll put it into my software that I want to, out of 100 compounds, I want only these 10 compounds of a specific molecular weight I want to detect. So it will detect only those 10 compounds, not the 100 compounds, on the basis of their mass to charge ratio. And there is a electron multiplier tube, which will enhance the signal. So generally, if you see the cost of, and if you see the efficiency of a particular mass spec, it depends upon the quadrupole and the mass analyzer. So there are different mass analyzer like time of flight, ion trap, orbit trap, and the quadrupole. So it has a different application. So in case of biopharmaceuticals and for proteins and peptides, they use a HRMS, that is high resolution mass spec. For toxicological studies, for pesticide analysis, for contaminant analysis, we use a quadrupole, that is a targeted quantitation. So how it works? So like my sample is getting eluted from the column in case of LC. Then there is ion source. There will be a formation of ions. So the difference is like in case of LCMS, the ion source is at atmospheric pressure, whereas in case of GCMS, it is under a vacuum. So that is why it is called as in case of LCMS, it is called as a atmospheric pressure ionization. So in this case, the ionization will happen at the atmospheric pressure. Then there is an interface to vacuum where your ion guide will move your ions towards the mass analyzer that is a quadrupole, which is completely in vacuum, and the detector. So the sample introduction, ionization, separation, and the detection. So there are different atmospheric pressure ionization techniques. So there is electrospray ionization, then atmospheric pressure chemical ionization, atmospheric pressure photoionization. I will not go into the deep of this since we have a limitation of time, but I'll just let you know. I'll not go into the principle of it. Yeah. So it is like what I was saying is like electrospray ionization or hazy. So in our company, we call it as a hazy, that is heated electrospray ionization. So if you see the large number of compounds, like most of them mid-polar to polar compounds are ionized by this ionization technique. Whereas APCI, it is for non-polar to mid-polar compounds, and APPI is purely for non-polar compounds. And if you see the molecular weight range also, so in kilodaltons also, so like proteins and peptides, right from the small molecules to high molecular weight compounds, ESI is applicable. So as you can see through the figure, ESI is the most used ionization technique so far in case of LCMS-MS. If you compare with the GCMS, so GCMS is specifically for non-polar compounds, that too with low molecular weight. So if you see the application of GCMS as compared to LCMS, so LCMS has a wide application, wide number of compounds can be detected by LCMS-MS, whereas GCMS has a little applications are little limited, as the limitation is there. It detects only non-polar compounds and which should be in volatile in nature, and the molecular weight range is at a very low level. So I was talking about the ionization source. So this is the ionization source where the ionization will happen. So this is your probe, and in this space the ionization will happen. So I will not go into the deep of this. Then I'll talk about the mass analyzer. So we have seen the LC part. What is the function of LC part? So I'll again summarize it. Like when we inject a sample, we want to detect a different compounds. So to detect a particular compound, we need to separate those compounds through a column, that is a stationary phase. After that, it will go to the detection thing. So when we are using a mass spec, so it will go to the ionization chamber where the ionization will happen. And after that, it will go to the mass analyzer, which is the heart of your mass spec. So this mass analyzer, what it will do, it will detect a particular mass. So there are different mass analyzer, as I mentioned earlier also, that is a quadrupole analyzer, time of flight, ion trap, orbit trap, and hybrid analyzers. So hybrid and traprid, like when we combine the quadrupole, orbit trap, and ion trap together, it becomes a hybrid or tribrid machines. So how the quadrupole looks like, we'll focus on more on quadrupole because we are interested in contaminant analysis, toxicological data, and the pesticide residue analysis. So the majorly the analyzer which we use is a quadrupole. So this is how it looks. So when you how it filters is like there is a RFDC voltages which will allow only specific ion to pass through the quadrupole, and all other molecules will hit to the rod. So like here there is a resonating ion and a non-resonating ion. So resonating ion will flow towards the detector and non-resonating ion will hit to the detector. So here also you can see into the video kind of thing, GIF image, and it happens in milliseconds. So this is how the quadrupole works. So as I mentioned, there is RFDC voltages which will allow only selected ion pass through the quadrupole. And these are the detectors, so EMT and PMT. So what is the function of these detectors? To enhance the signal. So when you, so like contaminant analysis is like a very trace level analysis, like PPB and PPT level. So that is the reason we need a mass spec to identify those compounds. And after the ion is detected, we need to amplify the signal through a series of diodes, and that is done by electromultiplier tube or photomultiplier tube. So yeah. So you might have heard the term single chord MS or a triple chord MS. So why it is important that people are using a triple chord MS, and what is the advantage? So when you have a single chord MS that is having only single quadrupole, so in this case we can do a full scan, and it will run through like through the mass range like 50 to 500. And you may predict that this peak is this chromatogram, and this peak is coming at a very giving a good sensitivity. So you may predict my compound of interest may be this one, but you can see a lot of baseline noise is there. It is not straightforward to interpret this data. In case of SIM, you will select a particular ion. So you will select a single ion and you will try to identify the compound. But the amount of organic impurities and looking at the complexity of the matrix, there are chances that different organic compounds may have a similar molecular weight. And in that case, your machine may misinterpret the results, and it may show a different compound as that particular ion. So that's the reason there is a role of SRM or MRM. So SIM is selective ion monitoring or single ion monitoring, where SRM is selective reaction monitoring or multiple reaction monitoring. So here what it does, it detects the parent mass. So that is a molecular weight plus charge, like maybe in case of LCMS, we see M plus H or M minus H. So if I have a compound like 200 molecular weight, so I will see 201 in M plus H positive polarity. So Q1 will detect the parent mass. In Q2, the fragmentation of that particular parent mass will happen into daughter ions, and in Q3, the detection of the daughter ions will happen. So the accuracy is more. So if I have only single chord, so there may be different compounds which is having a similar molecular weight, and I may misinterpret the data. So that is the beauty of the triple quadrupole in collision cell, that is Q2, there is a fragmentation, and Q3, there is a detection of that particular product ion. So every compound may have a similar masses. Means some of the compounds may have a similar masses, but very less probability is there they will have a similar daughter ions also. So that is what the advantage we take of triple chord here. So I'll not go through this slide. Yeah, so this is the ion path. So once your compound or a solution is eluted from the column, it will go to the ionization. So here the ionization will happen. After that, there are different, so a lot of R&Ds are going on in industries, in different vendors, wherein maximum number of ions should be entered to the quadrupole. So there are different patents are available for different vendors, how they allow maximum number of ions to allow into the quadrupole. So this is like an ion guide and the neutral blocker. So to get the maximum sensitivity, we need to get rid of the neutrals as well. So that neutral blocker is also there. And after that, it will go to the quadrupole one, which will detect the parent mass. Then it will go to the Golgi cell, where the daughter ions or fragmentation will happen for that particular parent mass. And here there will be detection of those daughter ions and the detector where your signal will be enhanced. So this is a maintenance part, so we'll not go into this. So different applications are there for LCMSMS, so like multi-residue pesticide analysis in food communities, then multi-residue pesticide analysis in beverages, multi-residue pesticide analysis in confectionery products, aflatoxin analysis, antibiotic and weight drug analysis, and so on. So I'll focus on food safety. So there are different applications of LCMSMS with respect to food safety. So some of the key facts about the food safety is like food safety refers to the proper handling, cooking and preservation of food in order to protect the people from foodborne illnesses. So the unsafe food can be like due to harmful bacteria, viruses, parasites, chemicals, which causes more than 20 diseases ranging from diarrhea to cancers. So this is a very interesting fact, and this is from WHO. So an estimated 600 million, so almost 1 in 10 people in the world gets ill after eating a contaminated food, and the death rate is 420,000 a year. So that is very alarming, and especially the children kids which are under 5 years of age, they are more prone to these foodborne diseases, and closely 1,25,000 deaths are there every year. So this is a WHO data. So the food contamination, if you see overall, it can be categorized into three things like physical contamination, chemical contamination, and biological contamination. Physical contamination can be like when any foreign object comes into your food, like hair, glass, metal, paste, jewelry, dust, fingernails. Whereas chemical contamination can be pesticides, herbicides, veterinary drugs, microtoxins, and so on. So our role is like to detect the chemical contamination using the sophisticated machines. Biological contamination, again, this COVID is going on, so this can be due to the viruses and the bacteria. So that is a biological contamination. So what is our role is like in chemical contamination. So there are commercial testing labs, government labs, then there is a regulatory bodies which decides the maximum residue level for a particular contaminant in a particular matrix, and the validation. So validation is like when you develop a particular method to detect a number of pesticides, we need to validate. So there are different guidelines to validate the method. And there are manufacturers like us who provides the high-end machine, and there are analysts, researchers, and scientists who develop the method, who runs the sample, who acquire the sample, and after that they review the data and finally report out. So the testing, chemical testing, can be categorized into two ways. So wet chemistry. So generally we don't use a sophisticated instruments and like total protein estimation or nitrogen content, for example, Geldal method we use. Then there is analytical and advanced analytical. So analytical is like HPLC, UHPLC, UV-visible spectroscopy, FTIR, and ICP. Whereas advanced analytical is like mass spec, all mass spec, like LCMS-MS, GCMS-MS, ICP-MS, Orbitrap is also mass spec, fusion, and everything. So what we do with this is like, why there is a need of this sophisticated instrument is like, these contaminants are at trace level and PPB, PPT level. And if you see the MRLs of FSSAI, that is Food Safety Standards Authority of India, and again the UA MRLs, these are all very trace level, like 10 ppb, 5 ppb, and to detect those compounds at that level, we need to have a advanced analytical tool with us. Like normal HPLC won't suffice the work here because it has a detection capability up to ppm level. So the regulation, so I'll just quickly go through this. So in India, we have a Food Safety Standards Authority of India, that is FSSAI. Then European regulation is there, and Chinese regulation is there, then American, that is Codex. And if you see, if you go through this link here in the Google, so if you see this, you need to just type the particular matrix or maybe the commodity, and you will see which are all compounds or contaminants needs to be monitored as per European regulation. Whereas this is for FSSAI. So if you see the FSSAI document here, this is the contaminant name 24D, and these are the matrix commodity where we need to monitor these many pesticides. These pesticide, and these are the maximum residue limit. So this is in ppm. So it is like 0.05, that is 50 ppb, and so on. So different matrix will have a different MRLs for a particular pesticide in different regulation. So what we need in the lab to start the analysis. So for example, if I had to start the pesticide residue analysis or veterinary drug analysis in my lab, so what are the things we require? We require first of all the mass spec with the LCMS. If we want to go for pesticide analysis, then supporting equipments like centrifuge, vortex, and consumables, then the column, so which is a stationary phase, then the reference standard. So since this is a targeted quantitation, we need a reference standard. So why the reference standard are required? Because we'll plot a linearity, and against which we'll quantify our unknown samples. So for that, we require a pesticide standard. If you want to do a wet drug, so wet drug standard, then solvents. So again, liquid chromatography, so methanol, water we need. And this is required, this is specifically for pesticide residue analysis. This is a sample preparation. So this is a Raichur's method we are going to discuss in my next slide as well. This I will not go, this is a LCMS method. And totally, as I mentioned, it's a 10-minute method. So in 10-minute method, we can analyze close to 300 pesticides. So different extraction protocols are there. So this is specifically for pesticides. So this is a AOEC protocol. This is like original Raichur's method, then AOEC method, and the buffer Raichur's method. This is for European. And the different steps are there. So like we need to take a 15 gram of sample, then we need to add a 15 ml of acetonitrile, then we need to add a Quacher salts like MgSO4 and 1.5 gram of sodium acetate. So what happens here, like we are able to separate the organic layer from the aqueous layer, and all the contaminants we are expecting into the organic layer. And if we are using some complex matrix, like when we want to find pesticides in chili powder or milk or chicken, which is very fatty, in that case we need to use a cleanup material. So the basic funda is like how much clean your sample is, the perfect way you can quantify your samples. Otherwise there will be a lot of matrix effect, peak shake will not be good, and you will not be interpret or quantify your samples perfectly or accurately. So these are the steps. So this is divided into extraction and the cleanup. So the Quachers is like quick, easy, cheap, effective, rugged, and safe. So previously, like 10-15 years back before, there was a lot of solvent consumption was there to prepare a particular sample. It was very laborious, and it was not like reproducible, and it was not also safe because we are using a lot of solvents. But now we are using only 50 ml of solvent to extract those particular pesticides from the matrix. So, and it is very quick also. And we have been seeing a very consistent result using this Quachers method. So that is why it is called as a quick, easy, cheap, effective, rugged, and safe method. So similarly, for that was for pesticides. Similarly, we have a different protocol for veterinary drug analysis through LCMS-MS. So I'll not go into much deep of this. So here are some examples. So like when I want to prepare a tomato sample for pesticide analysis, so this is how after sample preparation. So all the pesticides, all the contaminants we are expecting on the top layer, that is organic layer, acetonitrile layer. So this is in tomato, this is in cucumber, this is in chili powder. So in chili powder, you can see dark color extract is there. So in this case, we require cleanup material. So MgSO4, PSA is to remove the amino acids and organo acids. So that is a primary, secondary amine. Then C18 is to remove the fats, and GCB is to remove the pigments, that is graphitized carbon black. So this is for chicken. So chicken, fish, and lamb to determine the contaminants. So as we know, like these are all fatty matrix, but non-pigmented. So we'll use C18 material, not the GCB here. So these are the actual photos from our lab when we did the validation in different matrices for pesticides and other antibiotics and everything. So this is in okra, spinach, wheat and rice, chloramphenicol in honey. So that is a very nowadays very hot topic, like chloramphenicol analysis in honey, because it is having a very less MRL, that is 0.3 ppb. So this is like in honey and milk. And I was talking about the validation guideline. So this is like a Sante guideline. So I'll not go into much of these, but these guidelines are there, which will help us and to validate the method what we develop on our machine. So these are some of the application notes. So these are the ready to go solutions. So if you have a LCMS in your lab, and if you want to start the analysis. So for example, if I want to start the analysis in identifying and quantifying the pesticides in apple and grapes, so you can directly take this method and you can start the analysis. So right from the instrument method, sample preparation method, which are all pesticides we have detected, everything is mentioned in the application note. This is a part number. This is available through this link as well. So similarly, this is in chili powder. So everything is done in our lab. So most of the time you might have seen people shows the data which has been acquired and the method is developed in European lab or USA lab, and that may not be reproducible in India. But here, all the methods which I am showing are, we have done it in our lab. I personally, along with Dr. Dashrath, we have done it this in CSC Ghaziabad lab. So this is in rice, that was in wheat, and this is in rice. This is in cucumber and tomato. This is in apple and apple juice, single residue molecule, that is diethionone. This is in spinach and cabbage, multi-residue pesticides. Then this is chloramphenicol in honey. And this is like multi-residue pesticides in meat matrix, like chicken, fish, and lamb. And this is a very interesting method So conventionally, people have a separate method for antibiotic analysis, separate method for pesticide analysis, separate method for aflatoxin analysis. Even the sample preparation method is different, instrument method is different. But what we have come up with here is like single extraction method, single instrument method to detect pesticides, veterinary drug, and aflatoxin M1. So in milk. So why I have taken aflatoxin M1 only because it was done it in milk. So this is a single method, single extraction, single instrument method to detect pesticide, veterinary drug, and aflatoxin M1 in milk sample. So I'll not go into much of this. This is a software. So this is like we have a ready-made compound databases available with us for different compounds. So this is the software part. So this is how we identify and confirm the identity. So there are a lot of parameters associated. So when you inject a particular sample, so it is not, it's very simple to interpret the data and confirm the identity of a particular molecule. You need to go through all the data, like we need to match the ion ratio. So what I told about the validation guideline of Sante. So we need to see everything. If everything passes, then only we can confirm the identity of a molecule in the sample. So that's it from my side, sir. Thank you so much for your attention. If any questions are there, I am happy to answer. Thank you.
Yeah, thank you. Your wonderful presentation, and this is most important area which you have explained about the LCMS and how it can be used for the tracing of the different quantification and contaminants in food matrices. Definitely, there will be a couple of questions from our audience. There is no doubt at all. My appeal to Ramesh, shall we finish the the presentation from your probably Sarvendra, who is going to do next, so that we can come back for the question and answer in the last session, if you're okay, or you can finish as you come in.
As per your wish, sir, I'm okay with both the things, no problem.
Okay, so who is going to next, Suresh or Sarvendra, so that I can make them co-host?
So my colleague Sarvendra will start with the GCMS presentation now.
Okay, Sarvendra, I think already Sarvendra made him a co-host. Okay, shall we start Sarvendra's presentation?
Yes, sir. Very good morning. This is Sarvendra.
Yeah, Sarvendra, so that we can finish by 12 sharp, so that once the validatory completes, then we'll start our session, because the
Yes, sir. Surely, I am going to start my session right now.
Yes, sir. Because Sarvendra, we have 12:30. We have an important meeting with the PMO office to our Honorable Director of Education and Director of Extension. So kindly bear with for the inconvenience. So we'll start our valedictory at sharp 12 o'clock. So we can stop your presentation at 12 o'clock. Then we'll finish up the valedictory probably 25 minutes. Then we can continue our as per the scheduled program. Is it okay?
Yes, sir. Fine.
Yeah. Thank you. So now with us, Sarvendra P. Singh. He is a application development scientist from food and beverages. He has finished his Master of Science in Chemistry. Sarvendra is having more than 10 years of experience in the field of GCMSMS application development, and he is having a very vast experience in the field of food, beverages, and environment for the different toxicity applications or traceability applications in GCMSMS. He has extensive knowledge of method development for pesticides, veterinary drugs in various food matrices and validation. And today with us, he is going to explain about the GCMSMS, a tool for trace level quantification of contaminants in food matrices. Sarvendra, please, can you start your presentation?
Yes, thank you, sir, for the nice information, nice introduction. So I'm starting the session, and our topic is for GCMSMS, tool for trace level quantitation of contaminants in food matrices. So in my first slide, there you can see the agenda. It is, I'm going to cover the basics of GCMSMS, where I'll cover electron impact ionization, full stand mode of analysis, selected ion monitoring for the analysis, and SRM and MRM, SRM optimization, and GCMSMS applications. So here, my colleague Ramesh has presented lots of things for chromatography, how the separation happens. But I will relate those things to GC, and here I'll explain the front-end summary GC for the GC gas chromatography, Because in LC-MS/MS, you heard like HPLC is the front end where you need to go for the separation, chromatographic separation of the analytes in the mixture. But here, the group of components will be different, the category of components will be different, and we will be separating all the molecules one by one by using gas chromatography instrument. In this diagram, you can see it is already also a hyphenated technique, GC plus MS/MS. So in the GC, first of all, we need to inject the sample in the injector port, and then there is a capillary column, and that is placed inside the heated oven. And then we'll be providing the temperature gradient to the oven, and that will give the temperature to the column. And with the help of this temperature gradient, the elution of the molecules will be happens one by one, and we'll get the separation with the help of column. And then there is a heated transfer line, and this is the connecting link between the GC and MS. So the molecule, the column front end will be connected to the injector port, and then other end will be connected to the transfer line, and then the eluted molecules will pass through the transfer line and move towards the MS. So I will explain the MS in detail in my later slides. So I'm moving to the next slide. And here I would like to explain about the injector ports. Basically, that is the very important component of the gas chromatography system. So here in the injector port, it is a process of the vaporization of the sample. For example, if we are extracting analyte in a organic solvent, then we need to inject that extract with the help of autosampler, and the first touch of the extract will be go with the injector port. And here in the injector port, the temperature will be there, and with the help of that temperature, the extract will get evaporate, and the vapors will pass towards the column with the help of carrier gas. So in the GC, there is a mobile phase, that is a gas. If you compare with the HPLC, then liquid is the mobile phase in liquid chromatography. So with the help of carrier gas, all the molecules, all the evaporated molecules from the injector port will move towards the column that will be placed in GC oven. So here you can see the two diagrams for injector port. One is the most common injector port, splitless injector port. Here we cannot program the temperature. We have to place only single temperature, whether it is 200 degree or 250 degree, whatever you'll be setting the in the method, that will be the initial and final temperature port. But if you talk about PTV injector, that is programmed temperature vaporizer. So in the PTV injector, we can program the temperature. So for example, if some molecules are there, those can show some degradation at a higher temperature. So in this PTV injector, we can start the temperature from the lower side, from 50 degree or 80 degree, and then we can increase the temperature slowly towards the maximum temperature, and one by one all the molecules will get evaporated, and all the molecules will move towards the column with the help of carrier gas. So this is the column characteristics, and the column is basically a heart of GC system, and this is the main component that is responsible for the separation. If your column is not proper as per your required molecule, then that chromatography cannot get successful. So it is very important to understand what column is required for what kind of molecule. So in general, if I categorize the column, then there are wall-coated column, then solid support coated column, plot columns are also there. And basically our pesticide analysis is means multi-molecule pesticide analysis is dependent on this capillary column. We are doing the separation with the help of capillary column. So in the capillary column, there are three categories: narrow bore column, wide bore column, and mega bore column. Means this category is defined as per the internal diameter of the column. In narrow bore column, there is 0.1 mm ID, internal diameter is there. For wide bore column, 0.25 to 0.32 mm ID is there. And for mega bore column, 0.53 mm ID. See, on the basis of their internal diameter, the carrier flows we have to set. In narrow bore column, you can not set more column flow. In wide bore, there is also a limitation. And in mega bore column, you can go for 5 to 10 ml flow of carrier gas. In the table, you can see I have mentioned some critical columns, some important columns, those are very important for routine analysis. If you talk about, I placed all the columns in a increasing order of the polarity. So in TG1MS column, you can see it is totally non-polar because the coating is of only 100% dimethyl polysiloxane. And similarly, if we are moving towards the polarity, then TG5MS, it is less polar because only 5% diphenyl component is there, and rest 95% dimethyl polysiloxane is there. Then towards, if we are increasing more polarity, then 35% diphenyl, and the column is known as 35MS. Similarly, if you talk about the high polar, most polar column, that is trace TR FAME GC column for the analysis of fatty acid methyl esters. So on the basis of the analyte nature, if our molecule is non-polar, then we need to go with the non-polar column so that it can retain the molecule at the front side, and then with the increasing temperature, that molecule will get eluted from the column, and one by one, all the molecules will get separated with the help of this capillary column. And if we want to analyze, if we want to separate the polar molecules, then we need to go for the polar columns. So if we want to do the fatty acids, then this trace TR FAME GC column, that is more polar, and that will help you out for the separation of all the fatty acid methyl esters. So here I'm just introducing the mass spectrometry, and the mass spectrometry is the most advanced detector available across the globe. So here you can see the mass spectrometry that is working on the basis of mass to charge ratio. That is a basic principle of mass spectrometry. So for the analysis on the basis of mass to charge ratio, it is very much important to generate the ions. The production of ion is very much important. And the molecules, those you have evaporated in the injector port of GC, then you have separated those molecules with the help of capillary column that is placed in GC oven, and then they're transferred with the help of transfer line towards the mass spectrometry. So inside the mass spectrometry, there are few things like ionization of the molecules, and then ion optics are there for the movement of the ions, then quadrupole for the filtration of ions, and then detector is there for the detection. Now, if I'll explain this use of GC-MS-MS, where you can use this GC-MS, what kind of molecule you can analyze with the help of GC-MS-MS, then this particular slide will help you out that what kind of molecule you can analyze with the GC-MS system. So here, if you see the graph, then you can see if your molecule is very much non-polar and the molecular weight is not that much high, then you can go for the GC-MS-MS, and else for the high molecular mass, and if the polarity is increasing, then you have to go for the LC-MS-MS. And the different source are different ionization techniques are there for the LC-MS, that is ESI, electrospray ionization, and APPI, APCI. These are the ionization techniques available in LC-MS. And for the molecules, if we go, what kind of nature should be there for the molecules that you can analyze with the GC, then molecule should be volatile organic compound, because here your molecule should be in gas phase. If it is not in gas phase, then it definitely should get evaporated inside the injector port to convert in the gas phase. If the molecule is not volatile, then you will not be able to vaporize it inside the injector port, and that analysis cannot be happen. So it should be volatile organic compound, or it can be semi-volatile organic compound. At least some part of that particular analyte will get evaporated, and that can be transferred by the gas carrier gas towards the column. Thermally stable compounds, because this GC-MS technique is temperature based technique, and even in first stage, that is injector port, there is a temperature. So the molecule should be thermally stable. If it is not thermally stable, it will show you the degradation inside the injector port, and you will not be able to analyze that molecule. And it should be non-polar to less polar ranged compound, so that because polarity is directly proportional to thermal stability. If molecule is non-polar, then definitely it would be the thermally stable molecule. So these are the properties of the molecules that should be there if you want to analyze that molecule for the GC-MS-MS. And in the opposite phase, if the molecule is non-volatile, then you can go for the LC-MS. If molecule is more polar, then you can go for the LC-MS. So here, the system that is mass, I'm just introducing here that the total path of the ions that is inside the GCMS system. So first of all, there is a sample inlet, means that transfer line where you will be getting the gas phased ions and gas phased molecules, and then the ionization will be there. So for the ionization, there is an ionization chamber, and in the ionization chamber, ion source was placed. So inside ion source will help you out for the generation of the ions, and then the mass analyzer means the quadruples, those will filter out, those will scan all the desired ions, those you will be looking for, and then the detector for the detection of those ions, and then the data system. Data system means the software or your computer on the base with the help of that computer you will be recording the data, and you will be interpreting the data for the analysis. Here I will explain you the electron impact ionization, and this is basically a most common method for the ionization for GCMS technique. It is important, it can be useful for qualitative and quantitative both kind of analysis, and it is producing the mass spectra of the molecules, fragmentation it is giving. And then if you talk about the ion creation for electron impact ionization, then it is generally transferring the energy to a neutral molecule. That molecule should be in the gaseous state, and then it is ejecting one electron from its orbit, and then with the removal of that particular electron, the positive charge is getting developed on the that particular neutral molecule, and that molecule will get ionized. And after the ionization, because of one lone pair of electron, there is a free radical formation is also there. And as you know, the property of free radical is to absorb the excess of energy. So that's why, because the absorption of excess of energy, the fragmentation will also takes place. So in the GCMS, electron impact ionization is known as hard ionization technique, because it is giving the fragmentation of the molecules also. So for example, if you take this sample paper, that is molecule M with a molecular weight of 58, and then it is going towards the electron beam that is generated by the filament placed in the ionization source. And coming in the contact of this electron beam, one electron got knocked out, and there is a formation of positive charge and one free radical. So after this, because of this free radical, the fragmentation will take place, and you can see it is generating the fragment ions, that is 29, 27, 57, 15, and 43. These are the fragments, those are generated for this 58 number molecular mass. And 70 electron volt energy that we are using in general with the electron impact ionization, because this is the most suitable energy that will give the maximum yield of the ion, and that will give a suitable fragmentation of the molecular ion. If you see this diagram, here you can see if we are applying 20 electron volt current, then only a very little bit fragmentation you can see of the molecular ion, and the yield is only 10. And if you are increasing it for the 40 electron volt, then you can see some more fragmentation, and the yield is also increases up to 100. And most suitable, that is 70 electron volt, you can see the maximum fragmentation of this molecular ion, and the abundance is also 1000. So maximum abundance, maximum yield we can get with the applying of 70 electron volt current energy. Now we'll talk the versions those are available for GCMS mass spectrometry. Here two types of mass spectrometry is available: single quadrupole GCMS and triple quadrupole GCMS. In single quadrupole, you can see this ionization source is there, then ion transfer optics. This is S-shaped ion guide basically, and in S-shaped, it is helping out to filter out the neutrals, because this is here some RF and DC voltages are applied in this ion guide, and the neutral molecules will come straight and hit on this curve, and they will get lost. And our charged molecules, charged ions will pass out with the help of variable RF and DC, and it will move towards the quadrupole. And in quadrupole, there will be a filtration. There are four rods basically four electrode, we can say the four electrodes, and in two opposite rods there is a positive charge and in two negative charge, and on the basis of that variability of RF and DC, the filtration of ions will be there, and after the movement of this ion from the quadrupole, it will go to the detector directly. So in single quadrupole, basically we can do the full scan analysis if somebody is interested to know the molecules those are present in some extract, some herbal extract or some in plant extract. Then they can use simply the single quadrupole GCMS for the analysis, and detector is used for the capturing the ions, to converting those ions into the electrons, and then multiplication of the ions to generate intense signals that we can record with the help of software. In triple quadrupole, you can see the ion source, ion transfer optics are same. Quadrupole 1 is also same. But here you can see the introduction of this quadrupole 2 and quadrupole 3. So quadrupole 2 is basically a collision cell where one more time fragmentation happens. The ions, those you have filtered out with quadrupole 1, that will go to the quadrupole 2 for the fragmentation. In collision cell, with the application of nitrogen or argon, we can give some collision energy, and with the help of that collision energy, the fragmentation will take place. And then after that fragmentation, again the ion will move towards the quadrupole 3, and again we will be filtering, or we will be scanning the product ions now, and then those product ions will be detecting by the detector. So here the difference is, in quadrupole, in single quadrupole, you cannot fragment the ion further, and whatever ions are generated from the ion source, you have to detect only those ions. In triple quadrupole, you have the flexibility to fragment the ions twice, and here the precursor ion that you will be getting filtered from the quadrupole 1. Then again fragmentation will take place in collision cell 3, and then in quadrupole 3 you will be getting the product ion. So these are two systems I just wanted to show you, that is 7000 GCMS, that is single quadrupole, and 9000 GCMS triple quadrupole. The functions of single quadrupoles you can perform with the triple quadrupole systems also, but the specialty, that is the main feature, that is SRM, selective reaction monitoring, that you can do only in triple quadrupole. It is not possible with the single quadrupole. That is a main difference with the in between single quadrupole and triple quadrupole. Now I'll explain the scan modes, basically full scan mode and SIM mode. So if you talk about single quadrupole or triple quadrupole, in both the systems you can do the full scan analysis and the selective ion monitoring. You can do in both the both kind of mass spectrometry systems. In full scan mode, you have to select a mass range like 50 to 500, and then you can scan. Your system will scan all the ions; those are coming from the ionization source up to a range of 50 to 500, right? And this function you can do with single quadrupole, and you can do in triple quadrupole also. In triple quadrupole, you have the flexibility of two quadrupoles, quadrupole 1, quadrupole 3. In these two quadrupoles, you can select any of one that in which quadrupole you want for the full scan. And in SIM mode, you have to select that ion, that particular ion, those are coming from the ionization source. For example, 415, 417, 430, you have selected ions. So what will happen? Your quadrupole will scan only these three ions, it will not focus the other ions. So because of this proper ionization, because of focus on these particular ions, the sensitivity will increase, and lots of interferences, those are coming from the matrix, from the sample, those will get reduced. So selective ion monitoring, this way the selective ion monitoring is more sensitive as compared to the full scan mode. So if somebody is interested to know the unknowns, those are available in the some extract. So there is a NIST library that is giving around 3 lakhs of spectra of the molecules. So here, for an example, if you do the full scan analysis, then you can see this red spectra is the system generated spectra, and this blue spectra is the spectra from the NIST library. So simply you can do the search from the NIST library, and then NIST library will match the fragment, the spectra that you have acquired in your system, and then it will give you the probable result that what is the probability of this particular molecule that is you are analyzing, and you will be able to know that what is the molecule available in your extract. So this way you can see the example of acetylene. So after the full scan analysis, we have matched this acetylene spectra with this NIST library. And here you can see the proper fingerprinting of the spectra. It is clearly matching. So I can say more than 90% probability of the acetylene presence in your extract.
Dr. Sarvendra.
Yes, sir.
Yeah.
Yes.
So we'll stop your presentation for a while. Then we'll come back for your probably 20-25 minutes. Is it okay?
Yes, sir. Fine. No problem.
Our Director of Education is just joined because he has to attend another important meeting along with the PMO office. Right? Okay. No okay. So you can stop sharing for your presentation a while. Yeah, please stop sharing.
Yeah, I stopped it, sir.
Yeah. Thank you, Sarvendra, for your wonderful presentation. We'll continue with the remaining speakers after 25 minutes or 30 minutes. Our Honorable Director of Education, sir, is going to address with us, along with the Director of Extension, sir, and along with our Dean, sir, is going to address for this in the afternoon valedictory section. That is, it's going to start shortly, maybe another 1 or 2 minutes. And also joined with us our 10 days webinar co-organizing secretary, Dr. Uday Kumar, sir. So we'll start. Device, sir, we'll start the program, sir. Sir, you can unmute, sir. Just yes, sir. Yes, sir. Unmuted, sir. Sir, we'll start the program, sir.
Okay.
Okay, right, sir. Okay. Yes, sir. So now we are with a 10 days. Sir, before that, D, sir, will start the program, sir. D, sir. D, sir. Chitapur, sir. Hello. So we are going to start the 10 days webinar valedictory session. It is online virtually. Respected Honorable Vice Chancellor of the University, Dr. K. N. Kattimani, sir, who has inaugurated this 10 days webinar on role of nanotechnology in food and agriculture on 20th of this month. And it was continued successfully for 10 days with the morning a well-learned speakers, research experts in the field of nanotechnology. And almost 10 speakers, they have completed their different area of nanotechnology in their specialization. And in the afternoon session, we have almost 20 different industrial instrumentation application specialists, sir. So they almost all the sessions what we have planned in the morning as well as in the afternoon without any change in the schedule, we have successfully completed. We have another 2 speakers as going to complete probably after the session. We're going to complete that particular expertise in the field of LCMS and GCMS and SRMS. Today we have in the inaugural session, our Honorable Vice Chancellor has inaugurated along with our Director of Research, Dr. B.K. Desai, sir, address our August body of more than 1,107 who has officially registered for this 10 days webinar, sir. And in that, as per you can see the male and female ratio of this particular webinar, 56% male and 44% female are present in this particular webinar, sir. And now we are witnessing a valedictory session of 10 days webinar, sir. We have our Dean of the college, Dr. U. Satish Kumar, sir. May I request U. Satish Kumar, sir, for address this welcome address for our today's valedictory session. Satish Kumar, sir, please.
Thank you. Respected Director of Education, sir, and the Director of Extension, and all the participants of the this nanotechnology webinar. At the end of the 10 days program, as Dr. Sharan Kumar has stated, successfully has completed, and many have get benefited and get educated in the many of the participants regarding the application of nanotechnology, which is the very important aspects of the in the domain of agriculture also in the recent days, and that is a right time that many of them can benefited out of the nanotechnology applications, especially in agriculture. In this regard, that successfully completed 10 days program has got credentials to the college, and on this occasion of a valedictory function, on behalf of the college and on behalf of the organizing the webinar, I heartily welcome to Honorable Director of Education for this valedictory function, sir. Welcome, sir. And I also welcome Director of Extension also. He may be joined shortly. I extend all of the heartily welcome to him. And I also welcome all the participants who have been participant or enthusiastically beyond 1,000. It's a credit to the college. I welcome all of you. Thank you.
Thank you, sir, for your sweet and short welcome address. We have with us already Director of Extension, Dr. B.M. Chitapur, sir, is joined, sir. So, sir is on the line.
I welcome him on behalf of.
Thank you.
Yes, sir. Yes, sir. Yes, sir. So once again, I welcome to all the officers of the University of Agriculture Sciences Raichur and Board of Management of the University of Agriculture Sciences Raichur, and all the fellow participants and the organizing secretaries, organizing coordinators. I heartily welcome once again for this valedictory session. So may I request our Director of Education, Dr. M.G. Patil, sir, to address this valedictory session. Sir, M.G. Patil, sir. Sir, please.
Thank you, Dr. Sharan Kumar. Is it audible?
Yes, sir.
Okay. Good afternoon to one and all. Guest of honor, the senior most experienced faculty member, Director of Extension, Dr. B.M. Chitapur, sir, Dean, College of Agricultural Engineering, Dr. Satish Kumar, Organizing Secretary, the young proactive scientist, Dr. Sharan Kumar Hiregaudar, and another young enthusiastic professor and head processing and food engineering, Dr. Nidoni, faculty members of agricultural engineering Raichur, distinguished speakers and participants. As I know that more than 1,000 participants have registered for this webinar. I think this is a new type of webinar. So I think a lot of participants have shown the interest, and the ratio between male and female as indicated by Sharanagowda Hiregaudar is almost equal. So I take this opportunity to congratulate the organizing secretary, Dr. Sharanagowda Hiregaudar, for organizing this successful, excellent webinar during this pandemic COVID-19 period. I am happy to be the part of this valedictory program organized by Center for Nanotechnology. As indicated by Sharanagowda, the webinar duration was 10 days, and there were 3 sessions daily, morning, afternoon, and evening. Morning sessions, the lectures were delivered by the renowned scientists of National and International Institute of India and from India and abroad. Afternoon sessions were delivered by experts and application specialists from various multinational companies. And evening technical session was reserved for application of different instrumentation used in nanotechnology. So this nanotechnology is a new science, especially it is in agriculture also, especially for quality food production. The nanotechnology is applied in agriculture right from the seed. Right from the seed treatment, the nanoparticles are being used, and also for plant protection, the nano sensor. I have seen the one plant protection guest lecture was on sensor that is being used in management of the insect pest, and also the plant pathology. So as a result of that, even the bio nanoparticles are also being used in agriculture to enhance the yield and also quality of agriculture produce, and also for sustainable production or sustainable agriculture. And right now, not only from seed to harvest, even in post-harvest also, the nanotechnology is being used for value addition and for package. Application of the Center of Nanotechnology of US Raichur is doing an excellent research in the field of nanotechnology for agriculture, food and agriculture. And as I know that nearly 27 master's students and the 9 doctoral students have completed their program during a short period. I think 7 to 8 years, the Nanotechnology Center has been opened in US Raichur. Within this short span period, nearly 27 master degree and the 9 doctoral degree candidates have completed their degree program. Apart from this, 4 technologies have been already gone into the package of practice for which are helpful to the farming communities. And 3 patents are there for their credits. So the center is also offering a course on nanoscience and technology, its application for master and doctoral degree. So in this way, the center is going yeoman service to the farming community and the students, and also it is giving a very good name. We are getting a very good name for the UAS Raichur. This 10-day webinar certainly benefit our young scientists and the postgraduate students, especially after the completion of this seminar or outcome of this seminar. I think our young scientists can rethink the research in nanotechnology, and even good research programs can be formulated for postgraduate and doctoral degree students. So I intend to express my concern to all the participants to make better use of this outcome of the webinar in their profession, to glorify the name of their institute from where they have come and the country also. I once again thank the organizing secretary and his associates for successful completion of 10-day webinar on role of nanotechnology in food and agriculture. Thank you.
Thank you very much, sir, for your excellent elaborative valedictory address for the 10 days webinar hosted by the Center for Nanotechnology. Thank you very much, sir. Now with us our Honorable Director of Extension, Dr. B.M. Chitapur, sir, to address the gatherings. Chitapur, sir, please. Sir, please unmute, sir. Please unmute. Sir, unmute, sir. Yes, sir.
Thank you, Sharan Gowd. Good afternoon to all. I think I am audible now.
Yes, sir.
Sir,
Okay. I am happy to be part of this webinar, which has been inaugurated by Honorable Vice Chancellor Dr. K.N. Kattimani, who is so enthusiastic. And also I am happy that today I am with our Director of Education, Dr. M.B. Patil, the knowledgeable person in the university. I also I'm happy to be with the deans, and especially I'm more happy to be with the special invitees who have delivered lectures, then faculty of food science and technology, faculty of engineers, students, and the more I can specifically say a few words about Dr. Sharanagada, who has taken so much interest in organizing this 10 days webinar. It is not just one day seminar, webinar, 10 days webinar, and he was he could able to get the specialists from abroad, as far as USA, Italy, New Zealand, and also from across the country, Mumbai, Chennai, and Kolkata, and other places. For his efforts, I appreciate his efforts and congratulate congratulate him and his faculty for the way in which they have organized this webinar, and they could able to also do it successfully. I am happy, as our Director of Education said, the university is very young, just a decade old, and the faculty of food science and technology is still younger than the university, but still with lack of men and material, with limited staff, they could generate so many new budding scientists, so many theses have come. Apart from that, particularly this nanoscience technology, nanoscience division, I can say, they have come up with many useful technologies. They have many patents to their credit. Now I came to know that they have developed a nano material for preserving vegetables. Then I know that they have also developed a material which can reduce the use of pesticides, particularly insecticides, and they have worked on some instrument which can help in purifying water in a lesser time, more efficiently, and which has been actually implemented in one of the villages. In that way, they have done a lot of work. Besides that, they are holding this webinar now, a webinar across the nation they are holding, and in which more than 1,000 participants have participated. Really, it's an astounding effort, I can say, and I really compliment him once again. As far as nanoscience is concerned, it's a new science. It's a new science, but science which is making a very rapid progress in the field of, not only in the field of other engineering or IT sector, particularly in agriculture and food science. In fact, it is a science which controls the processes that are happening with a much at a smaller speed. They have been stimulated, or they have been controlled efficiently by using these nanomaterials. In that way, it is really more beneficial to the human race, I can say, particularly in the field of food science and agriculture. We are really, I can say, we are benefited, because for the country with so much population, 1.3 billion population, and nutrition is another problem. We say poverty, hunger, and all these things. Under such things, this science has a role to play, and to deliver that, our scientists could able to bring the stakeholders, all the other faculty members and particular students can be given some view, some vision, and some thoughts for their future growth. In that way, they have really done a marvelous job. And this technology, science, nanoscience is not that old, I said. And during 2016, as many as 14,000 documents have been documented, and about 2,800 patents have also been patented in this science. So it is a rapidly developing science, and it is also taking more of the market also. I learned that the market, which was just 110 billion in 2002, it is more than 1,000 billion now. So it is also grabbing the market because of its importance. And many say that nanoscience or nanoparticles are toxic. Some say that nanoparticles are toxic, but you see the signage and the attitude of scientists. They have come out with green nanotechnology. They have come out with green nanotechnology, which can help carrying out process safely, which are energy efficient, which are which have lesser residual toxicity, and which are also from the point of greenhouse gas emission. And as far as food science and agriculture is concerned, many chemicals have already come into the market. US is leading, but India is not far behind. I have come to know that there are materials, particularly in case of this fertilizers, we have nano green, nano max NPK fertilizer, tag NPK fertilizers like that. 3 of the products are being produced in India itself. And in pesticide and herbicides also, there are many chemicals. And one most beautiful thing or most useful thing that has happened is, I think it is from Germany, who have produced these nanoparticles of gamma omega 3 for consumption, so that which can help the patients suffering from heart problems. And many of the nanoparticles materials, particularly vitamins, can be directly taken through mouth, and it can help the nutrition of people. There are nanometals which can help in storage, particularly protecting from bacteria and actinomycetes or many other disease-causing organisms like ZNO nanoparticles, Ag nanoparticles, and even in some of the nanoparticles are also useful in packaging. Particularly, they have been developed for packaging the milk and meat, which help to know the oxidation process or discoloration process like that. In that way, this field has a really vast area, I can say, in which one can work, and this can really help the humanity. For that, whatever the efforts made by the scientists of our university is commendable. Along with our vice chancellor and also director of education and director of research, I compliment their work and also commend their work for completing this webinar, 10 days webinar successfully. Finally, I hope that in long term, this innovations in the field of nanoscience are going to help the human race, particularly in their nutrition. With these words, for providing me an opportunity to share my views with you all, I thank the organizing secretary and also the faculty of food science and technology and the dean of College of Agriculture Engineering. I thank you and wish all the best to all the participants. Before that, I really commend all the invitees who have delivered really commendable lectures. And this is one science wherein the private and public together are doing a much better work for the humanity. Thank you. Thank you for your time.
Thank you very much, sir, for your excellent coincidence between the particles and the across the global level what the scientists are working with the nanomaterials, not only the material science, which is also included with biomaterials, that is green nanotechnology, what you have explained, and in the consideration of the farming community. So as we already know that the nano zinc and nano boron and nano phosphorus is probably it is going to take in the future days, sir. Definitely the nanoscience and technology is going to help the farming community in the future, in the due course of time. Thank you very much, sir, for your sharing your thoughts in the field of nano science and technology, and appreciation for well-learned speakers as well as the scientists who are involved in the field of nanotechnology. Sir, it is a small information to the both of you, just another 1 or 2 minutes. So almost 10 research experts from the different parts of the country, and 20 different industrial experts, and 18 different parts of the states who have been participated in this 10 days webinar, sir. And in terms of the country, there will be one USA, UK, then New Zealand. In the morning, we have a New Zealand speaker. He has explained about the application of nanotechnology in encapsulations, and one is from the Italy, and remaining experts from the different parts of this country, sir. So almost we have covered all the areas pertaining to the role of nanotechnology in food and agriculture, like encapsulation, like surfactants, like basic applications, by antimicrobial properties. Then in case of instrumentation, all the advanced instrumentations like even XPS, Raman, then SEM, then TEM, all the instruments which they have explained. And today we have a toxicity experts for the nanomaterials. So after this, again, this valedictory address, we are again going to probably another half an hour or so, we are going to continue this particular session because of this emergency meeting, sir. So on behalf of the organizers, on behalf of the college, and on behalf of all the fellow participants who have joined with us in this valedictory session, so we are extending a sincere thanks to our Honorable Vice Chancellor, who has given an opportunity for us to conduct this for 10 days webinar, sir. So thank you very much, sir. And also in the valedictory session, we have with us our Director of Education, Dr. M.G. Patil, sir, who has addressed to all the participants and made the day a grand success. Thank you very much, sir. And we have our Director of Extension, Dr. B.M. Chitapur, sir, always man behind with us for taking the technology into the farming community. So thank you very much, sir, for your valuable presence and addressing to our all the participants. Thank you, sir. And we have with us our Dean of the College, Dr. U. Satish Kumar, sir, who has welcomed the August body, and also our Dean, Dr. M. Yurrugoda, sir, and our Head of the Department, Dr. Uday Kumar Nidoni, sir, who is the co-organizing secretary of this particular 10 days webinar, and all the coordinators of this organizing team, Dr. M. Nemichandrappa, sir, Ayingoda, sir, Maheshwar sir, Polisoda, sir, Sushilendra, sir, Prakash, sir, Vijay Kumar Palle, and all the my department faculties who have joined with us for this 10 days program. So I extend my sincere thanks to all the members who have joined with us for this valedictory for 10 days webinar. Thank you one and all, sir. We are going to start continue with this webinar for the remaining session, sir, with your permission. Thank you very much, sir. Dr. Sarvendra P. Singh, are you with us?
Yes, sir.
Yeah.
Yes, sir.
Shall we continue the session for another half an hour or so, with your permission?
Yes, sir. Sure, sure.
Yeah. Thank you.
So welcome again. And we were talking about the selective reaction monitoring, SRM mode. Okay, so this is a advanced mode for the scanning for mass spectrometry systems. And here, the best feature, that is, we can say the advancement in mass spectrometry, and we known as it is a triple quadrupole technique. And here I explained that total 3 quadrupoles are there: quadrupole 1, quadrupole 2, and quadrupole 3. And quadrupole 2, basically it is a collision cell where further fragmentation happens and product ion formation will be there. So in selective reaction monitoring, SRM mode, you can see in Q1, the precursor ion was selected, that this ion will go only towards the collision cell for the further fragmentation. And in Q2, you can see then after fragmentation, another few ions was formed. And then in Q3, one product ion among all these formatted new products was scanned, and it moved towards the detector for the detection. So if you see this slide, then here in quadrupole 1, you can see the selection of precursor ion. In quadrupole 2, again the fragmentation of the precursor ion, and in quadrupole 3, the product ions, one product ion you can see it is highlighted, that is selected, and in the detector you can see the peak generation, the signal recorded by the detector. And here if for the quantification, we are generally taking the area of this particular peak for the calculation of the results. So if you see this top diagram in Q1, lots of ions are coming from the ion source, and in the quadrupole 1, one ion got selected, 2 ions got selected. And in quadrupole 2, again fragmentation of these 2 ions, and in quadrupole 3, one product ion was getting scanned and moved towards the detector. So this is the basically the feature of triple quadrupole system that you cannot do with the single quadrupole techniques, because for the fragmentation and product ion scanning, there is collision cell and quadrupole 3 requirement is there. So now, if you see this diagram, then I have summarized the data from the full scan mode, and then selective ion monitoring and selected reaction monitoring. So in full scan mode, generally we are scanning range of ions. So here you can see, you cannot see the proper peak, proper signal that that the detector has been recorded, because it was looking for all the ions, and the time window was really very short. So it could not be focused on all the ions at a very high sensitivity. But if you talk about selective reaction monitoring, where we have selected only few ions, 2 or 3 ions, then you can see, if we see, look at the M by Z 182, then you will be able to see the signal of that 182 ion. But as you know, this is coming from the source directly, and there can be some more ions of 182, that those are coming from some more molecules. So that's why you can see some other peaks also here. Those are also coming for 182, but for the different components. But here in the SRM mode, if you see this, this is a transition basically, 182 to 153. We have selected 182 as a precursor, like we have selected 182 in SIM mode. Here also we have selected 182 as a precursor, and then further fragmentation happens in the collision cell. And after that, 153 product ion was formed, and then we have scanned that 153. So here, there is more selectivity. You can see that we have scanned a particular transition, and now you can see a full-fledged peak here, signal that we have recorded, and lots of background got removed, and this peak is very much Gaussian and very much quantifiable and very much identifiable. It is because of this selectivity, it is coming towards the sensitivity, that it is reducing the selectivity in sample preparation. Because if you talk about the complex matrices like food matrices or herbal matrices, these are having lots of color pigments, lots of matrix ingredients. Those are other than your desired molecules. So in this kind of thing, you don't need to think like this sample is simple and this sample is this matrix is simple and this matrix is complex. You can just go ahead with any kind of matrix, and you can reduce lots of matrix background, lots of other interferences, and you can get your desired signal properly. And for a single quadrupole or full scan, you need to have some analyze some higher concentration of the molecules, and you'll not be able to get that much of selectivity. And that is the consolidated GCMS method due to high performance. Compressed chromatography is also possible here because of high selectivity here. And this gives a easy data processing. You don't have to see for other peaks that what is there and what is that. So it is giving a single peak cut to cut, so that you can easily identify and you can easily process your data, and you can report the results. Now, if you talk about the analytical methodology that is related to the application, then first of all, if you are having any matrix, then you have to see what is the requirement that you have to do for that particular matrix. So for the requirement, lots of regulatory guidelines are there, like FSSAI, European Union. FSSAI stands for India, European Union stands for the European countries. Those are giving the MRLs, maximum residue levels of those particular molecules. Those are like toxicants, and that can give some bad effect to the human bodies, human beings. And then getting those requirements from the regulatory bodies that X molecules is required in that particular matrix up to this level. So we need to know that what is the requirement and what you want to do for the analysis. So after getting those requirements, you have to go for the sample preparation procedure. You have to prepare a sample. Preparation means just you have to extract the molecules those you want to analyze. So after extraction, the molecules will come in your organic solvent, and you will be placing that extract in a auto sampler GC vial, and then you will be putting that vial to the chromatographic system. And then in the chromatographic system, like gas chromatography, after the injection, chromatographic separation will happen. And then with the detector, that is mass spectrometry, in detector, you will be getting the detection, and with the help of software, you will be processing the data that has been acquired from your instrument, and then you will be reporting the results. So if we talk about the food safety for the pesticide analysis, basically, then this is some details from the validation guideline that we need to see if you are working with the food matrices for the validation of pesticide molecules with GCMS or LCMS. So here you can see we are talking about unit mass resolution, that is mass spectrometry, MSMS, and single quadrupole we are talking about. So in single quadrupole, minimum 3 ions are required for the targeted kind of quantification, and in triple quadrupole, minimum 2 ions should be there for the quantification. And the signal to noise ratio should be more than or equals to 3 for any kind of LOQ you are defining for the method. And ion ratio is also a very much important criteria for the identification and confirmation of that molecule, because if you are having 2 ions, then you will be getting some difference in the areas intensity of those ions, and there is a correlation of ratio. So that ratio will become your ion ratio, and that ion ratio for entire batch for that particular molecule should not be more than plus minus 30%. For example, if you are injecting X standard and it is there is a 50% ion ratio between both the ions that you have selected for the analysis of X. So if the ratio is 50%, then for other samples it should not be varied more than plus minus 30% of the 50%. So this is the requirement of identification and confirmation for the molecule that you will be analyzing with GCMS-MS. And if you talk about the performance parameters, then there should be a linearity calibration curve you might have aware. And for the calibration curve, minimum 5 levels are required to inject. And in all the levels, there should not be more than plus minus 20% variation if we are calculating back that particular injection with the help of linearity. Matrix effect, as you all are aware, that food matrices are really very much complex matrix, and lots of other ingredients are there. Those can give some interference. So that is known as matrix interference. And because that matrix interference will contribute in the suppression of the intensity of that ion, or the enhancement you will be getting for the intensity of that ion. So it is very much important to them to understand the matrix effect, like a simple matrix where there is no other ingredient like your other than your molecule. Then in matrix effect, then you can quantify those particular molecule with solvent. The external standard is spiked in the solvent. But for the complex matrices like food or herbals or like that, then you have to go for the matrix match calibration curve, where you'll be compensating the matrix effect that is coming from the matrix. LOQ, limit of quantification, it should be less than or equals to MRL. Specificity, it should not be more than 30% of reporting limit. Recovery, that should be 70 to 120%. Recovery is basically the known concentration that we are spiking in the sample, at the earlier stage before the sample preparation, and after the analysis of the data, we need to see that what amount of that known concentration you are recovering, that amount should be in a range of 70 to 120%. Precision, if we are injecting multiple replicates of that particular recovery, then we need to see the percent RSD, and that should not be more than 20%. Robustness is also means reproducibility and reportability kind of parameter, where we are verifying the validation. Ion ratio, I have explained it, and retention time. Retention time is basically the time, at what time, up to what time the molecule was retained inside the system, and that time is known as the retention time, and that should not be more than plus minus 1 minute for each and every molecule. So here I will talk about this application. I have taken an example of rice sample, and we have followed the Raichur's AOAC 2007.01 method. Raichur's means quick, easy, cheap, effective, rugged, and safe. So basically in rice, if you talk about the nutritional content, then 10% of the moisture is there in rice. It is really very less moisture content that is not suitable for this method. 80% of carbohydrate, 7% of protein, very less fat, 0.66%, and sugar is also very less, 0.12%. If we are going for the extraction, then we have taken 5 grams of sample in 50 ml extraction tube, that is a centrifuge tube. For the recovery, we spiked at this stage for 5 ppb, 10 ppb, and 50 ppb. Then as per the method, we have we added the 50 ml of HPLC grade water that was also acidified with acetic acid, and content was 1%. Vertex it for 1 minute and soaked it for 10 minutes. Then we have added 15 ml of acetonitrile, 6 gram of MgSO4 and 1.5 gram of sodium acetate we have added. MgSO4 we are using for the removal of excess amount of water from the sample, sample solution, and 1.5 gram of sodium acetate was used for the adjustment of pH from 4 to 5 range. Then again vertex it and centrifuged it at a 5000 RPM for 5 minutes, and after centrifugation the cleanup stage will start, and the supernatant has been cleaned with 150 mg MgSO4 and 50 mg PSA for the cleanup. So this amount is enough for 1 ml of supernatant. If we need to clean further 2 ml or 5 ml, then accordingly we need to increase the amount in the multiples of the ml. Then again vertex is for 1 minute and centrifuged at 10,000 RPM for 5 minutes. Cleaned extract was placed in GCMS vial and then injected to the instrument. This is a method that we have followed for the GC and MS condition. On the basis of these parameters, the system will acquire the data. And this is the total ion chromatogram that we have observed after the acquisition of data. Here 150 plus pesticide molecules was there in the mixture, and this chromatogram stands for the 10 ppb, and here you can see the very sharp and resolved peaks. And if you talk about the early eluting and late eluting molecules like alidochlor and deltametrin, they are also giving a very prominent peak shape. So this way we can say that chromatographic separation is very good coming from the system. And if you talk about the LOQ and linearity, then you can see here this calibration curve plotted for 5 calibration points, and the coefficient of correlation, that is R square, is 0.9971. And for R square, the criteria should be more than 0.99. So it is passing the criteria, and we can say we can quantify the results with this calibration curve. And if you see the detection and the sensitivity at the lowest level, that is 5 ppb, then it is also very much prominent peak and well intense for the quantification. If we talk about the identification, then here you can see this fluoride-4-p-butyl, the M by Z was 91, and it is a very good peak. And then for the quantification, we have used this ion, and for the confirming ion, 238 ion number was used. And as I earlier mentioned, that minimum 2 ions should be there for the targeted analysis of pesticides in the GCMSMS system. So here you can see the 2 ions proper, the quantitation ion and the confirming ion. And between these two, there will be an ion ratio. And here you can see at the bottom side, the ion ratio range was calculated directly from the system automatically. And the range was 62.49 to 116.04. And it is coming 89.29. That ion ratio is coming. So it is falling in between the range of this ion ratio. And that's why we can say this is the confirmation about this particular fluorG4-p-butyl molecule. And it is confirming and identification is proper for this particular molecule. And next we can go for the other results for this particular molecule. Here, if you talk about the retention time that I have mentioned, that it should not be more than plus minus 0.1 minute. So you can see the retention time for Winclose-Align of quantifier and retention time. RSD of retention time was really very much outstanding, and you can see it was 0.05. So that is also very much better, you can see. Now, if we talk about the processing window that is coming from the software. So in this window, we have lots of features that we are utilizing. Here you can see the compound list. We were having the list of 150 plus molecules, but here in a single window, it is not possible to show. So that's why limited molecules you can see here in this screenshot. Then this is the quantitative ion. On the basis of quantitative ion, the results will be quantifying, and the values those will be coming, that will be on the basis of quantifier ion only. These are the qualifier ions, means confirming ions. On the basis of these confirming ions, we'll be confirming that presence of the molecule, and the ion ratio should pass for these two confirming ions also. These are the customized flags. If you are setting a requirement that if the requirement is passing, then the flag should be green, otherwise it should be red. So that's why in the blank, there should not be any presence of the molecules, and definitely there is no presence. You can see NF is there, not found. So that's why the flag is red. And for the molecules where the presence is expected, so the flags are coming green. It means all the parameters like RSDs and recoveries, ion ratios, RT, everything is getting passed. That's why the flag is coming green. So no need to worry about to see data in depth. This flag will inform you that whether some deep analysis is required, or you can go in a single shot. Now, if you talk about the ion ratio, yes, sir.
Yeah,
yes,
sorry to interrupt you. So can you make a little fast so that we can cover the other speakers? I think other speakers like our Suresh and Subodh.
Yeah, only Suresh is there, sir.
Okay, how about Dashrath?
I think he'll not be available. Okay. No, he may join in between the session.
Fine. So right we are with the Suresh, right?
Yes, we have only Suresh, and I am also having 2 to 3 slides more.
Please. Thank you.
Yeah. Now I am explaining the results here, and for the RISE analysis, the ion ratio, the area repeatability, recovery, and precision, I'll show you here. So in the RISE for the molecule cyclorate, the ion ratio RSD was 5.6%. As you see, it is very much straight graph that is coming for the ion ratio, and upper limit was 75% and lower limit was 40%. So all the ions were giving the ion ratio in between this range. If you talk about the area repeatability, then also you can see there is not much variation in the areas of these particular molecules, and area percent RSD was also less than 10%. For recoveries, it should be in the range of 70 to 120%. And you can see for all the molecules, the recovery is coming in between the range of 70 to 120%. So we can say that our method is very much suitable for the analysis of pesticides, for the selected pesticides in the RISE, that is giving the results within the range of 70 to 120%. And if you talk about the RSD, because we have injected 6 replicates for each recovery point, and here you can see for 10 ppb, the percent RSD was less than 20%. This y-axis is starting from 0 and maximum is 20, and you can see all the molecules are falling under this range. So if there is any incurred presence of the pesticide in your sample, then how we can get to know that what amount is present of that particular molecule in the sample. So here I have demonstrated here with the calibration curve technique. For chlorpyrifos ethyl, you can see the calibration line is starting from the above than 0. So here definitely there may be some presence of the particular molecule. And then we have used a different calibration technique, that is standard addition calibration method. And in a standard addition calibration method, that you can see now line is coming from the behind the 0. And the calculated concentration you can see on the top, that is 15.013 ppb, that is coming for the chloropyrifos methyl in the rice sample. So this is an example of the real application of this particular method for the analysis of chlorpyrifos methyl in the rice sample. So if you check and compare it with the regulatory requirements, those are like MRLs for rice. If we see the MRLs of rice for chlorpyrifos methyl, in European Union, requirement is 0.5 ppm, means 500 ppb. In Japan, it is 100 ppb. In China, 500 ppb. In India, it is 500 ppb. So we are getting only 15 ppb. So we can say your sample is passing the criteria and that sample is not failed. So although the presence is there and it is beauty of method, beauty of system, that we can able to know that 15 ppb is there, but it is not passing the criteria of the regulatory limits. So we can say the sample is passing the criteria. So this is all from my side, and if there is any question, I'll be happy to answer the questions.
Yeah, thank you very much, Sarvendra, for your nice presentation on GCMS, a tool for trace level quantification of contaminants in food matrices. As I just mentioned with even Ramiz also, so that we can have a question and answers session at the end, so that we can finish on time. So
no problem, sir.
Yeah, is it okay? Is it okay?
Yeah, yes, that is fine, sir.
Yeah, thank you.
Thank you, sir. Thank you.
Thanks. So can I have Suresh? Suresh is with you, or I'm making to co-host?
Yes, sir. Suresh is there.
Suresh is there. Suresh, in which name he has logged in? Did he log in?
Yes, Suresh, can you hear us?
Suresh? Because I can see M Suresh, I think is not M Suresh, right? Is it
M Suresh. M Suresh.
Okay.
Yes,
yes. Okay, thank you. So I made him a co-host right now. Suresh, can you hear me? Suresh?
Yeah, hello.
Yeah, Suresh, can you hear?
Yeah, now it's.
Yeah, because I made into as unmuted for all, muted for all. So now I made it to you a co-host so that you can unmute yourself. Yeah. So we have with us Suresh Murugesan. He is also a application specialist. He is going to explain about the ICP-MS, a tool for trace level quantification of metals in food matrices. So basically, Suresh is a MSc analytical chemistry. He did his master's, and he's having more than 10 years of experience in elemental analysis with QA, QC, and the method validation skills using ICP-MS, ICP-OES, AAS technologies in various segments like food, beverages, and finished products. So basically, he's working with the food and agriculture and even beverages also. On request, all the team members have joined with us for this explanation of these quality parameters by using the LCMS or GCMS, now with us ICP-MS. May I request Suresh, can you start the session? Yeah.
Thank you, sir.
Yeah.
So,
Suresh, how much time you required?
yeah, I will take on 25 minutes to 30 minutes.
Yeah, you make it at least 25 minutes. Thank you.
Yes, I will try this. So please confirm the screen is visible.
Yeah, fine. Good.
Yeah, thank you. So here, my colleagues Ramesh and Sarvinder has explained about the high-end equipments like LCMS, MS, and GCMS, MS. So in continuation with that, there is a ICP-MS, which is called inductively coupled plasma mass spectrometer. So which is used for the analysis of elements at a trace levels. So this agenda, the screen is not moving. So,
Can you share it again? Sometimes
yes.
it may be stuck. Yeah.
Yes.
Yeah. Fine.
Yeah. So we will see about the introduction of the technique, and then the working principle and hardware of the ICP-MS and food analysis workflow, and then we will see the applications in food safety. So when we talk about this ICP-MS, as we know about, this is the technique used for the multi-elemental analysis at the trace levels. So we have the concern like what is the need of measuring the or quantifying the elements in our food and agricultural products. So as we all aware that the elements, heavy metals like lead, cadmium, arsenic, mercury, these are all causing they are very toxic to the human body, and this causes certain specific issues with the uh, certain internal organs, like it is shown here. Ladies brain, making the damage for brain, kidney and central nervous system, because they are having the capability to accumulate in our tissues, so they will cause the toxicity of the to the human bodies. So there is a lot of sources to this to the contamination to our food and our beverages as well as the agricultural products. So this is the requirement. Sorry, 1 second. So this is the requirement for the sir. Can you confirm that, please?
Yeah,
I
yeah,
am
yeah.
audible.
Yeah,
Yeah.
you're right.
Thank you. Sorry. So this is the
Suresh, you are unmuted.
yes, sir. Just for I'm. Hello.
Yeah.
Sir, sorry, I'm just changing the place. The network issues there.
Okay, okay, fine.
Yeah. So this is the issue for the related with the heavy metals, and we have to monitor their level in our food materials which we are consuming. So as per our regulations, FSSI as well as the international regulations such as European Union and US FDA, they have the contaminants regulations for metals, and that is a concern we are thinking about the quantification. And they are even at the trace levels, they are causing very impact on our negative impact on our health. So it is a necessity to monitor these elements. And as we know, with our nanotechnology nowadays, we the nanotechnology is utilized for the as a preservatives, food additives, and food packaging, food processing issues. So most of the metal oxides are used for this process and as a nanoparticles. And at certain amount, it is as per the regulation, if it is exceeds the limit, then it will be a reason for a toxic effect in our body. So this is important. At trace level, we have to monitor that, so that we have the different technologies, like here you can see that atomic absorption spectrophotometer and ICP OES, like the emission technique based, and then mass spectrometric based indirectly coupled plasma mass spectrometer. So the flame atomic absorption spectrometer and the graphite furnace atomic absorption spectrometer working based on the absorption technique, and the emission technique based is the indirectly coupled plasma. ICP-OAS we call it as, or ICP-AES, and ICP-MS is mass-based technology. So these are all certain, predominantly used technologies in the laboratories to decide and quantify the elementals which is present in our food. So here, what is the difference between why we have to select ICP-MS instead of these technologies? So the main thing is comes under the the capability or the sensitivity of the instruments. So I am showing here the difference or the dynamic range and the sensitivity of the instruments. You can see here the AAS, which is having the sensitivity around sub ppm level. A graphite furnace is having the very good sensitivity when compared to the AAS and ICP-OS, but the process which is used in the while doing the heating of the graphite furnace, it takes the temperature program. It takes a lot of time to get the fixed temperature, and because of that it takes a lot of time, and it is a sequential technique. So one by one element we have to measure. So every time for if you are measuring 5 elements, then you have to take one by one elements. You have to fix lamp and you have to analyze. So that takes a time. ICP-OS is having a very good multi-elemental technology, and it can be measured over a range with a good dynamic range. Okay, around a ppb level 2, it can detect sub ppb level 2 greater than percentage level, we can detect that. So ICP-MS is having very it's also the multi-elemental technology, and simultaneously we can detect almost all the elements present in our periodic table, and which is having very good sensitivity at lower concentration, as well as it covers almost higher concentrations. So nowadays the people widely utilizing this ICP-MS for the the trace level analysis, as well as it is giving the high dynamic range up to 10 orders, greater than 10 orders. So this is widely accepted technique in the field of trace elemental analysis. So here, this thermo is this is worth mentioning here. From this, based on the sensitivity and the all from the AS to ICP-MS, single quadrupole ICP-MS and triple quadrupole ICP-MS. Apart from that, high-end equipments like sector field ICP-MS and multi-collector ICP-MS, the every range of product, it is covered in the thermoscientific technology. And so this is ICP-MS ICAP RQ model, which is we are going to see about that single quadrupole. So this in general, inductively coupled plasma mass spectrometry covers main actually that 4 parts, that is ICP and a sampling interface and ion focusing optics and a mass spectrometer, ion detection. So let us see that how they are working and their hardwares in a fast manner because of the time consumption. ICP is a source, which is, we know this ICP-MS is working based on the ions which is produced in the plasma is separated by the mass spectrometers. Mass spectrometer is the quadrupole technology, which is separating the ions based on the M/Z ratio. So the ions we need to be generated. So the ICP is utilized for the plasma generation. We are using the argon gas for generation of plasma. The plasma contains around 6,000 to 10,000 Kelvin temperature, and which is produced by the coupling activity of our argon and the radio frequency. So that creates the plasma, and it is having very high temperature, and the when the sample is aspirated to this plasma, it gets the ionization, and that ions are focused towards the ion optics and then mass spectrometric region. So here, this is the process when you aspirate the sample. So the sample containing your elements, which is in the molecular form, like in a if solid form, so that will be dissolved and brings back as a metal, as a solid, and then it melts, and then it is gets vaporized, so that it produces the gaseous atoms, and then the actually gaseous molecules are produced. Then they are breaking into the atoms, and then they are ionized. This is happening within 5 millimeter distance between the torch, which is producing the plasma, and the cone system, that the sampling interface. So then it enters to the cone system, and why you are using that argon gas instead of there are a lot of another gases we can utilize for that, but the argon gas is very important, and it makes that sense that the ionization energy of argon is 1500 kilojoules per mole. In electron volt we can say that 15.8 8 electron volt. So that is almost giving the ionization energy of almost all the first ionization energy of elements will be below than this limit, and few noble gases are having high energy than ionization energy than this argon. But if we look at the second ionization energy, they are very high than the argon gas. You can see the graph. So the second ionization energy is higher than this argon. So it will be forming that doubly charged species of our elements. The thing is, if the mass spectrometry is having the detection based on m by z, so mass by charge ratio, if the charge is increased, the complexity of the analysis will be increased. So the organ which is producing that singly charged species, so it is very helpful to directly detect that m by +1 charge species. So it is simplifying the the detection of our and the quantification of our elements. So that's why we are using that argon gas as a predominant gas for the generation of plasma. So next one is sampling interface. This is actually the plasma is generated in the atmospheric region, and they are ions which is generated in the plasma which is going to the mass spectrometric region, and the mass spectrometric region is kept in the vacuum, high vacuum, around a 10 to the power of -7 tar. So there is a cone system which maintains the vacuum, as well as they are acting as a interface between this atmospheric region and the vacuum region, and they are actually sampling the ions which is entered to the mass spectrometric region. So we have the sampler cone and skimmer cone. The sampler cone is containing 1 millimeter orifice, and then further we are having the skimmer cone that is having a 0.5 millimeter orifice, and then we have the ion optics, so extraction lenses. So this optimizes the sample interest, and also it is actually maintaining the acting as a barrier between these two atmospheric regions. And then it is they are actually sampling the ions. So the ions which is entered to the high sensitive mass spectrometric region is protected. So then we have the quadrupole system. You know that already Ramesh and Sarvendra has explained how the quadrupole is working based on the applied RF and DC voltages. So I'm not going too much about this. So this is working based on the mass to charge ratio. So just a simple graphic illustration to understand how if we are passing the multiple ions, how they are separating, filtering out the ions, and that how the detector is receiving. So you can see this blue color and orange colored dots, which is showing that multiple ions which is entering to the quadrupole. Based on the applied DC and RF voltages, they are allowing one by one, at a particular time, the only a single mass to charge ratio based on that voltage set of the DC and RF voltage set. It will be allowed to the detector. Then it reaches the once the filtration happens based on the mass to charge ratio, they reach the detector. Here we are using the secondary electron multiplier tube, which is working based on that Einstein's photoelectric effect. Okay, so the ions which is hitting the diodes, and they generate the secondary electrons, and they are multiplied through the array of the diodes, and they are giving the signal. Okay, and here we are using the pulse count for the lower concentration, and then higher, if the concentration is high, the ions are high, so the ion current mode, we call it as analog mode, is utilized for the detection. So this is the hardware. I want to simply explain that with this model ICAP RQ ICP-MS. So we have the sample introduction system, which contains peristaltic pump to suck the sample from the auto sampler, or if we are doing the manual analysis, it is actually applying the pressure to suck the sample, and it is aspirated to by the nebulizer, and it is aspirated to the spray chamber. The spray chamber is kept under the Peltier cooling. It is called the electrical cooling. The temperature will be 2 to 3 degrees centigrade. So that the bigger droplets and the finest aerosols will be separated out with the help of the cyclonic action and the the surface conductive. Due to this Peltier cooling, the condensation will happen, and then fine aerosols around 10 micrometer will be separated, and it is passing through the torch. So torch is generating the plasma, where the 10,000 around 10,000 Kelvin temperature is generated, and the aerosols, which is having very finest size and containing our elements in the molecular form. So they are ionized in this torch, that is exactly in the plasma, and the ions are allowed with the controlled manner by optimizing their flow with the help of the interface system, which is containing sampler cone and skimmer cones. Then we have the rapid lens. This is the this is the ultimate thing here, which is actually deviating the ions which is coming from the sampling interface. So here, why this is kept here? Because the ions are deflected with the electrical and magnetic field. So the neutrals and other particles are not reflected. So photons and neutrals are generated in the plasma that need to be removed, so that we will get high high sensitivity and to avoid the background. So this right angle positive ion deflection lens, which is reflecting the positive ions which is generated in the plasma, and it will remove all the neutrals, so that we get the very good sensitivity. Then we have the Q cell. We called Q cell here, that is collision and reaction cell, where we apply the helium gas or oxygen and other gases such as hydrogen, ammonia. These gases are utilized for removal of the polyatomic interferences. The polyatomic interferences are generated by the it's with the our atoms and molecules which is containing multiple elements, which is produced in plasma itself. So it need to be removed to avoid the false positive detections. So we incorporate this Q cell, which is effectively removing that polyatomic interferences along with the low mass cutoff technology. It is a patented technology, and that avoids the further the precursors for forming these polyatomic interferences will be avoided further after this Q cell also. Then the ions enters to the actual filter, that is the mass analyzer. They filter out based on the M by Z ratio, and they enters to the detector. And we have seen that the signals are by the multiplication of electrons, they are detecting that as a signal, and we are getting that using the readout machine. We are getting the intensities as a final result. So in our as actually this analysis, ICP-MS analysis, we are doing this is the workflow we can call it as. So we have to prepare the sample, which is converting the homogeneous type of solution to aspirate to the instrument. Then we have to confirm that the day-to-day analysis for we have to confirm the performance of the instrument before doing our regular analysis. Then the method need to be set up, and then the sequence, the prepared samples and standards will be arranged, and then the data interpretation will be once the analysis is performed by the instrument, then it will be with the help of this QTegra ISDS, Intelligent Scientific Data Solution, which is helping to simplify the user activity, and it is helps the data interpretation easier way and user friendly, so that it is helping in the data interpretation part, and so that we are generating the report for the final submission of the report, which is the quantification of our trace elements. So I am just showing the examples of our food matrices for this contaminant analysis, which is done in our Ghaziabad laboratory. So these are all the matrices with the different commodity groups. We can see here rice, wheat, which is comes under cereals and pulses product, and then milk, paneer, butter. This comes under milk and, or we can say, dairy products. So these meat products, chicken, mutton, these, and spices. There is a lot of spices which we are using for our day-to-day, food products. So and fruit and vegetables. So here are some examples which is obtained, the data obtained from the ICAP RQ. So we can go through that. And for the method performance of this analysis based on the regulatory requirements as well as the guidelines. So AOIC 2015.01 as well as the Eurochem guidelines for the validations. So it is summarized here for the understanding. So linearity, specificity, limit of detection and quantifications, accuracy, precision, robustness are established, and their criteria are verified. So the sample preparation, which contains that this is the actual process of sample preparation. It's a simplified way to understand the preparation. So this is generally for food and beverages. We use the microwave digestion system. It is having very good advantage over the conventional techniques like our open digestion system. So we require very less acid, very less sample size for the preparation of the for the ICP-MS analysis. So the sample will be, the homogeneous sample need to be weighed, and then we have to add the the process contains sample digestion contains oxidation process. So we are utilizing the oxidizing agents such as nitric acid, hydrogen peroxide, and some elements will be stabilized in chloride format. So we can include that hydrochloric acid, and then we are using that microwave digestion with the temperature program for the digestion. So what we are getting the clear solution, all the organic matrices will be removed from the compound. Food matrix contains carbohydrate, fat, proteins, all these organic material. They need to be removed from the stream, so that as I said in the rapid lens, they will create the background. So that need to be removed, so that this oxidation process making the efficient way, and they will remove that, and we will get the elements in the soluble salt format, nitrates, chlorides, sulfates, like. So those will be aspirated to the instrument, and they are getting the the mass spectrometer will help us to give the intensity even though at the trace levels. So these are all certain the materials and the process, the weight, volume, and the acid amounts. You can see that only very 2 ml, almost 2 ml to 4 ml only acids are used, and that preserves a lot of these acids are very trace metal grade. Even Optima grade acids are very high acid, means costly acids. We can save that, preserve that by optimizing these conditions. So these are elements which is mentioned in our FSSA regulation. Along with that, the ICP-MS is having the capability to measure multiple elements at a single aspiration. So I have selected a few more elements, around 21 elements, for our analysis in the food products, and the linearity is obtained with the very good R square value, as always, with the detection limits at very even PPT levels of detection limit you can see. So the intensity RSD shows that it is not greater than 10%, so that it is very good. Repeatability is assured with the linearity. Then it is several products, the rice and wheat, which is having, rice and wheat are comes under same commodity group, but they are differ in certain carbohydrate as well as sugar content, protein content. So slight variations in their nutritional value factor, so that the digestion and as well as the repeatability and the accuracy need to be ensured. So three level of spikes at three levels, the accuracy was ensured, and we have got but very good. You can see that if you see the cadmium value, it is 10 ppb. Actually, the FSSA requirement is 1.5 ppm, but we are getting at 0.01 ppm sensitivity it is giving. This is also with the spike and recovery, the actually the practical LOQs we can say, with very good repeatability. So like that we have measured in wheat. So for the time consumption, just we can see the data of accuracy and precisions for the different food products. So this is for wheat, and the accuracy at LOQ level. So I am concentrating on the lower side because the lower side is very that is important. So accuracy and precision at the LOQ level, which is mentioned that here. So as per that AOEC 2015 as a guideline, so if it is the concentration is less than 100 ppb, then it is acceptable at 60 to 115% of recovery. But still we are attaining at very far away from the 60%. We are attaining 80 to 108%. So it shows that the technique which is utilized for this method using the high-end equipment is giving very good quantification technology. So these are all again this is the milk and milk product, but here you can see that milk, paneer, and the butter product, these are all highly varying in their nutritional content, water content, moisture content, and fat content. So there should be a genuine the single method to analyze all the dairy commodities. So this is optimized, this method is optimized, and we have achieved the very good accuracy and precision in milk, and as well as that butter and cheese products. So it at a higher fat content also, we are getting 94 to 112% with this method using our ICP-MS technology. So this is for meat and meat products, chicken and mutton, as well as buffalo meat also checked with that for different varieties. So everything is giving as per as per AOEC, that is giving very good sensitivity, and you can see that the data, which is this is for the spices, and which is done with the turmeric powder, black pepper, and red chili powder. So this is food like fruit and vegetables. The okra and the tomato samples are utilized with the three level different concentration. Here you can see that actually for food and fruit and vegetables, 50 ppb level is the maximum limit. So we have this method, optimized method, is showing that at 5 ppb level of practical LFUs, this is spiked and achieved at 5 ppb level. So it is very good. Sensitivity is achieved with that. So this is the recovery in tomato fruit sample, and this is in the okra samples. And to ensure that the process, the data should be quality one, so we have to ensure the quality checks in the sequences. So it is with the help of the internal standard, we are checking the the interferences, like physical interferences. So it should be avoided so that we can get the high sensitive results. And that is with the help of the QTR software, we can monitor that, and we can define the criteria based on our regulatory things. And this shows that the red dotted line shows you that limits, and the actual achieved things are mentioned in the dotted way for different internal standards. So this shows that there is no deviation between our the matrix and our standard comparison, and it shows this interference free, or physical interference free analysis. Then this is that CCV standard. So in the sequence we have to ensure that after five samples, if something is going wrong, then it should be, if you are not monitoring that, then again all the results will be it should be ensured with that. So in the sequence we have incorporated the CCV standards, so that it ensures that sequence almost, you can see that 80 samples will be analyzed, and in between of 10 samples we have analyzed one CCV standard. CCV stands for continuum calibration verification. One of the standard is measured, and that ensures that your all the sequence is running properly and the data is quality one. So for further understanding of these methods, you can go through these application notes. I have given the links and for your reference for these above matrices. And there is a lot of applications is related with that. And as we said, we are talking about the nanomaterials. The nanomaterials are, they are contributing to different nano herbicides, nano material based fungicides, and the food processing and additives, which are using the metal oxides. They need to be, they will contribute to the elemental contaminations. Also
Ramesh?
we can get that. Yes sir.
Yeah, can you make it, because almost we are
Yes
running
sir,
out
I
of time,
have a few more slides only. I will complete that.
because you have taken
Yeah.
more than 35 minutes.
Yes, thank you.
Thank you. Thank you.
So this is just a one more coupling technology to make that. So in elemental cases also, there are organic and inorganic species are there. So at the different oxidation states, their toxicity level will be differs. So for that we need some hyphenation techniques which is coupled with our ICP-MS. So like ion chromatography, gas chromatography, liquid chromatographies can be attached with our ICP-MS, and then we can do the exact speciation studies for our different oxidation state of elements. So this is one of the example, and the Qtegra software supports that. The simple multiple software is not required when you attach the different IC or any other hyphenation techniques to the ICP-MS. So I think based on the time we are, I'm just giving the reference here. So for your reference as a food regulations, method analysis and guidelines. And this is our team, as already sir has introduced. So we have the team with headed by Dr. Dashrath and Dr. Brijesh Pandey sir, who is our business partner. So you can reach out our any queries related with our technologies and the applications. So thanks for your attention, and any questions we can take up now.
Yeah, thank you very much, Sarvendra. I think we have Subodh and Dashrath, they are in the line, I believe so. Shall I just check it? Subodh, are you with us? Can you unmute yourself, because I just checked in once, you were there. Okay, you just stop sliding. Yeah, fine. Dashrath is there? As I made him a co-host, probably he may left, I don't know. Yeah, so he's not there. Dashrath is not there. Yeah, Dashrath is there, no? Dashrath, I have made him as a co-host. Volkar, sir? Would you like to talk to about SRMS, Volkar, Dr. Volkar? Ramesh?
Yes, sir.
Can you just check it whether Volkar and...
Hello.
Yeah.
Yeah,
Sir. Yes, sir. But how much time is there? That is my concern.
No, the question is, now it is almost running out of time. Like now it is 1:28. Okay.
Yes.
So probably 15-20 minutes. Is it okay? How about Budukoti is going to Subodh? Whether he is going to...
No. Subodh and Sarvendra has presented.
Okay.
Sure. Because we are mainly three instruments we have focused:
Correct,
LCMS,
correct.
GCMS, and ICP-MS.
Correct. No, that was excellent. There is no doubt at all.
Yes.
No, I just thought you are going to talk about SRMS. If it is so, we'll go ahead. Otherwise, we start the question and answer.
Yes, sir, because I thought that it's a time taking and 15-20 minutes is not enough, sir.
Okay.
So,
Okay. Then,
yes.
then we'll do, Volker sir, we can have a question and
Question.
answer session.
Yes, yes, definitely, definitely. I am joining that
Yeah.
question. and answer.
In coming days, we can have your own.
Yes, yes, yes,
Yeah.
yes, definitely. I will add on that. No
Yeah.
issue.
Yeah. Thank you. Thank you. I think all three are in the online. Ramesh,
Yes, yes,
Suresh, and Sarvender. Okay.
yes.
We'll start question and answer session so that we have already a couple of questions in my chat box as well as in the YouTube and personal chat box. So probably we'll finish up maybe another 10-15 minutes. Okay.
Yes, sir.
Yeah.
No issue.
Yeah. So we'll start with Ramesh. Can we get portable type of detection of pesticide residue as a rapid method?
Yeah, so as I mentioned in my presentation also, like these pesticides are at, we need to detect at a very trace level. So the mass spec, what we see nowadays are becoming little short. So like initially when we see, it is like very big mass spec was there. But nowadays slowly the design is getting shorter and shorter. And even the HRMS, like our new HRMS, even it is shorter. But the portable one is quite difficult because portable is like few vendors have a GCMS as a portable device for online SP and direct cartridge. They will inject into the GC and they can do it. But in case of LCMS, till now that provision is not there. And one more thing, it requires a vacuum. So generally, when we do it like in HRMS or LC code triple MS, so it requires a vacuum. So portable, it is little difficult.
Okay. The next question. Yeah. Can GCMS be used to estimate the composition of unknown herbal formulation samples that are being used in natural farming? The natural formulations are prepared with wild weeds and other leaves of the plants along with the cow urine and cow dung. Is it possible?
Yes, sir. Actually, for this kind of samples, it is very much possible if you are extracting it with a organic solvent, and that extract you can inject on GCMS. But at that time, you have to work on full scan mode. And if you'll be working in full scan mode, then you will be scanning a range of ions, and whatever the ions you are getting for the different molecules or different analytes, those are present inside that extract. So with the help of NIST library, you will be able to search, and you can be able to know the probability of the molecules those are present inside that extract. That is possible, sir.
So we can do it, right? Yeah.
Yes.
So I have to do mercury detection in milk by using ICP-MS. So please share the protocol for sample preparation. What is the sensitivity of ICP-MS? Sorry.
Yes, sir. So mercury can be analyzed as already in the presentation we have shown about that dairy product analysis. So we have achieved around a 50 ppt level of LOQ. So it is possible. If you are sharing your, you can see that the application note for your reference, and that will help you for your work.
Correct. Definitely. Questions. Sir, what are the different most important sickness caused by misapplication of pesticides? Anyone can?
Yeah,
Can you
so
repeat, sir?
The question is, what are the different most important sickness caused by misapplication of pesticides? So while applying the pesticides, so we may fatigue or we may have some sickness. So what kind of causes or what?
So actually, as I mentioned in my slide also, these pesticides are responsible for wide range of diseases, starting from diarrhea to cancer. So different pesticides have a different lethality. So it may be worst and it may be a small little symptom sickness also. So that's the reason it is like some of the pesticide, maybe Dr. Dakshat can put some of the pesticide name which are very much lethal. So That's the thing. So basically, it ranges from diarrhea to cancer, and 20 diseases are there for the if you look at the lethality of pesticides.
Okay. What are the criteria one can follow while collecting the sample for residue analysis?
For sample, just Ramesh, I will add. For sir sample analysis, basically sampling method is there. So for fresh fruits and vegetables, there are sampling method, and if you want to go with the some cereal pulses or any commodity, there are European Union already published guidelines are there for sample collection. So based upon that testing requirement and based upon that matrix requirement, we can follow that sampling guideline as per European Union, and that can be done.
So based upon the sample requirement, we can change the sample protocol. Am I right?
Sample, no, we have to, yes, we have to select that what type of the matrix we are going to collect. Based upon that matrix description, we can say I will tell one example. If you are taking the grape collection, grape sampling as well as pomegranate sampling, so in grape sampling you have to collect that bunchlets, small bunchlets. Whereas you are going for the pomegranate sample, you have to select that individual units. Okay, so individual units for as per European guidelines, it is a near about 40 to 45 units you have to select. For in case of grapes, it's near about 6 kg sample.
Okay, it's depends upon the sample again, right?
Yeah. Yes, and another example I want to cite here. If you want to test as aflatoxin in peanuts, so there you need to collect at least starting from 2 kg to 20 kg sample.
Okay. Sir, we use the nitric acid, sulfuric acid, and perchlorate acid in the form of 9:2:1 digested plant samples to analyze zinc, iron, copper, and manganese in ICP-MS. What digestion method should we follow?
Yeah, here you can, this is the plant sample as you mentioned. It may contain certain amount of silicon with that. So this instead of perchloric acid and sulfuric acid, because this sulfuric acid is highly viscous and they will make some, the difference between your standards and your sample digestates. So I will suggest nitric acid and HCl in combination of, 1:0.1 like that. So that variation is, for example, 2 ml of nitric acid and 0.2 ml of HCl you can add, and 1 ml of hydrogen peroxide to support the oxidation process. So plant samples you can take 0.5 gram and dry basis. So you can process to the analysis. Microwave digestion is better for this.
Microwave, right?
Yeah.
Okay. This is a general question they've asked. They're just getting clarification. Tell me about the Premier Institute for Advanced Training on Nanotechnology in Agriculture. Do you have any idea about in India? So they want to get, okay, or else I can tell them.
Nanotechnology.
Yes, sir. Training on nanotechnology.
Okay.
Okay, no problem. I'll just put a word to them. So,
yes.
So in advanced training and nanotechnology in across the country, some of the institutes they are going to organize for 10 days, 21 days. But because of this pandemic situation, so ICAR has not given any kind of permission to the any training programs. But whereas in case of NHEP, so they have done a couple of institutes like Rauri, they have done the training program, and even TNAU is doing regularly, Kazri is doing regularly, Sirkot Mumbai is doing regularly. So definitely there are institutes. Even IISc is doing wonderful, not exactly the training, the research on nanotechnology, but in agriculture they had to start. Okay, so sir, ICP-MS is more sensitive than CVAS for mercury analysis in milk. Is it true?
Yes, definitely. It is actually the vapor generation can be the vapor generation technique for AAS released for AAS is at 5 ppb level you can detect at that, but ICP-MS you can go up to PPT levels of detection. So definitely it is good sensitive technology. The ICP-MS technology is a better one.
Okay. Sir, how many days do we store collected samples before residue analysis?
Storage.
Storage, sir.
Basically, that storage of the sample really depends upon the type of the pesticide. If that pesticide has too much volatile, then you have to do that immediately. If these pesticides are semi-volatile, or maybe say I can say it's non-volatile, then you can keep in freezing conditions, that to be in -20, so that molecule or concentration of that analyte will be remains same.
Okay, so I think
But those samples should be kept as it is intact form, sir, not after homogenization.
correct, correct, correct. Yeah. So I have done. If any questions or any clarifications, anybody wants to talk to our experts today, so you can raise your hands. I can connect them, or you can send me in the chat box through Zoom, or in my personal chat box, or in my YouTube chat box. You can connect because whatever the questions which you have already we have done. Anybody wants to add anything during the session? So please let me know before closing our session. Anybody wants to talk to with our experts or to me, so you can just raise your hands so that I can connect with you guys.
Sir, just I want to add one more point. You have asked one question now regarding some or unknowns identification.
Correct.
Okay, and this Sarvendra has clearly answered that through triple quarter basis. But there is one more possibility where we can utilize the high resolution mass spectrometer, and through that high resolution mass spectrometer, we
Sorry, you were not audible. Can you check your mic?
known as unknowns, and certainly we will get that unknown unknowns. Known as unknown we are considering because those are known to others, but we don't have their reference standard or certified reference material available with us, but we are taking the help of the advanced analytical tool, and we are trying to identify that compound is, yes, this is the probable compound, and based upon the references and those things we are trying to prove that yes, this identity is correct. Second thing is that totally unknown means that we have to do that lot of activity, and we have to come out with that proper justification, proper confirmation of the identity of that molecule.
Okay.
So
okay, sir, we have done almost from the chat box as well as and even in my personal chat box also. So
yes,
thank you very much. If I think Sarita Kumari has raised her hand, but I don't know whether she wants to talk to you guys. So let me check. Yeah, Sarita Kumari, I made you as a co-host. If you want to talk to anybody, if you want to ask any question, you can unmute yourself and you can talk to them if you are interested. Yeah. Sarita? Yeah, I don't know. Okay, it's okay, no problem. So there may be so many issues. Okay, so we have in the morning session finished by Dr. Parsarthi, has done a wonderful presentation on benefits of nano encapsulation. Then we have three speakers, Ramiz Ajad and Suresh Murugesan and Sarvendra P. Singh, who has done a wonderful presentation on different application, that too food traceable contaminants in food matrices on behalf of all the participants, on behalf of the coordinators, organizers, and all the fellow scientists who have spent their valuable time for this because sitting in one place almost 4 hours, it is not small easy task to everybody, and you have to maintain the battery power, you have to maintain the what you call the data in your mobile, and listening and interacting with the question and answer session. Really, I should appreciate to all the participants there for their sincere efforts, I can say, sincere efforts to understand, learn those who have given a knowledgeable information from the thermos. So I extend all the thermo people who have shown a very much interested, and really they have done wonderful presentation. Especially Ramiz was explaining about the LCMS on on the food matrices for whatever the toxic elements. And whereas Sarvendra was talking about the GCMS and Suresh was talking about the ICPMS, and even in the last, so Volker was explaining about so how we can, but in shortage of time, so really Volker as well as the Budukoti, so definitely we can have with you in coming days for the HRMS. So once
definitely,
again,
sir, definitely.
yeah. So once again, on behalf of the organizers, coordinators, and my below participants, I extend my sincere thanks to all the Thermo team for your wonderful presentation. Thank you very much. Okay, so we are end up with a 10 days webinar on role of nanotechnology in food and agriculture. So we started the day on day 1, that is on 20th of August 2020, by inaugural address about our Honorable Vice Chancellor, Dr. K. N. Kattimani sir, and guest of honor by Dr. B.K. Desai sir, and vote of thanks by our Professor and Head Dr. Uday Kumar Ndoni sir, and welcome address by our none other than the Co-Organizing Secretary Dr. M. Virnugoda sir. With this, igniting the inaugural session by the eminent personalities, so I can say with your support and with your blessings, so we have successfully completed 10 days webinar without any change of the schedule. So that is most important, without any change in the schedule. What we have made the schedule for 10 days, in the morning session, we have speakers of different experts. In the afternoon session, different experts from the renowned instrumentation. I would like to mention the companies like Thermo Fisher, and in that I should mention Dr. Arun. He has coordinated, Dr. Sheikh has coordinated for all the application specialists for this 10 days webinar. And also I should mention from AMIL, Biojet, who has deputed their persons, especially for the Biojet, Mark Freeman. So he has given application of the homogenizer, and from AMIL, as AMIL, and of course Carl G's, they have given, they have accepted, and AP Research, Stephen Proto has given the application specialist for AFM, and Pranab has explained about the XRD, and Vijay Joshi has explained about the TEM, and all the Thermo team, starting with Amrish Dave and Amit Joshi, and Dilip Rawat. He came twice for one is FTR as well as the Raman. Then alias, then of course the today's team, Ramiz, Suresh, and Sarvendra Singh, and Volkar and Subodhkar. So all the experts for 10 days webinar, without them, this program is not going to be conducted or organized, and it is very tough task to me to engage all of you and make into a grand success. And all the speakers, my gurus and my well-wishers who has given the different areas, I just mentioned few names like Prem Shanti, Dattatray Kadam sir, Anshup, Yunad Saharan, Nargun sir, Deepa Bhagat, Mohan, Deprasad Rai, Parthasarathi R, and Parthasarathi S. So all the 10 speakers have really covered a wonderful information and explained and interacted our participants very nicely, very comfortably. Thank you very much for all the speakers. Thank you very much for all the industrial application specialists for your wonderful time. That is most important. So I should say thanks to most important is on the day what we have on Saturday and Sunday, even all the participants. So it is crossed even Saturday more than 250 members, and even in Sunday also it is crossed more than 250 members. So that's what your spirit, that's what your involvement for knowing the information on nanotechnology in the field of food and agriculture. So definitely we can have our own network team, and any collaborative research work, if any scientist would like to do it, or any scientist analytical is required, and any kind of help is required, definitely we can join with you, because ultimately we are going to do for the development of the country and helping to the farming community. That should be our motto. So with this, there is no end, so we have to make it an end. So the what our well-wisher and our mentor, Dr. Nargun sir, was first slide was, it is like encouraging me. It is not nano, it will become a nano. So I should feel it is a nano. So from now onwards, you should feel yourself, I am also a nano. So that the Richard Feynman, there is a plenty of room at the, there is a plenty of scope at the bottom. There is a plenty of room at the space. So whatever the terminologies would they have mentioned, definitely it is going to be useful for your research work. So let us join our hands and say thank you, everybody. I extend my sincere thanks to everyone. Jai Hind, Jai Karnataka. Thank you. Jai Nano.
Thank you, sir. Thank you.
Thank you, Ramesh, and thank you, Sarvinder. Thank you, Suresh and Volker. So we are going to close the session, end the session. Thank you very much. See you soon.